So I think without further ado, we have the pre-doctoral trainees coming up first. And our first speaker is Delaney Fisher. Delaney comes to us from Vassar, and she entered the neuroscience graduate group. She has been in Elizabeth Heller's lab, and in fact is, I think, second or third author on a recent neuron paper entitled Chromatin-Mediated Alternative Splicing Regulates Cocaine Reward Behavior. She's also presented at a number of virtual, unfortunately, conferences since the last research day, an EMBL conference on epigenetics and chromatin, and the NIDA Genetics and Epigenetics Consortium meeting. She's also very active in outreach. She's co-director of the GLIA, which is NGG's outreach group, and she spends a lot of time mentoring undergraduates and folks in the Heller lab. And so we're really excited to hear her talk today on adding pieces to the puzzle, investigating the transcriptomic and epigenomic profile of the mouse striatum. Delaney?
Great. Thank you so much, Julie, for that introduction. That was really nice. Thank you all for tuning in today for my talk. I will share my screen momentarily. As Julie said, my title today is Adding Pieces to the Puzzle, Investigating the Transcriptomic and Epigenomic Profile of the Striatum Following Cocaine Exposure. And I just really wanted to point out the end of Julie's introduction that I do really enjoy mentoring, and actually the perm student who's currently a junior at Penn, Chloe Han, is on the call. So I just wanted to give a quick shout out to her that a lot of the work I'm about to present, Chloe helped me with over the summer. So the training grant has also really helped me improve my mentorship skills. So I'm really excited she was able to join us as well. So I will share my screen. Okay. Is everyone seeing the PowerPoint? Okay, perfect. Thank you. I'm going to make myself a little bit smaller. Great. Okay. So the Heller lab is a neuroepigenetics lab, and so we are really interested in how different epigenetic marks, such as histone post-translational modifications, might affect gene expression, and how cocaine can interplay with histone post-translational modifications. And so the overview of my talk today will be how I've been actually examining bivalency following cocaine. And so for the talk, we're going to go over what is bivalency, when did it join the literature, what is this mechanism I just said, the methods I use to examine bivalency, which is called sequential chip or chip-rechip, as some of you may know it as, some of my preliminary results, and future directions of the project. So in 2001, there came out this new theory called the histone code, or maybe not so new to some of you, by Genuine and Elias, talking about how really DNA is like the backbone of transcription and translation, RNA, protein. It's all about DNA access. And some of the things that really regulate DNA access are histone post-translational marks such as methylation, phosphorylation, simulation, ubiquitination, and acetylation. And for those of you who may not be super familiar with epigenetics, these marks all have different, sometimes opposing roles to each other, and we can find them on nucleosomes, which are made of histones where our DNA is wrapped around. And so a lot of these marks will either cause activating gene expression, repressive gene expression, and sometimes they might interact with each other. And so the basis of the histone code theory that was put out was that there are two factors that control how DNA is accessed with histone post-translational modifications. The first being the concentration of these marks. So how much methylation do you have? How much phosphorylation do you have on a nucleosome? But also the combination. So if you have an activating methylation mark and a repressing methylation mark on the same nucleosome, what happens? And so drugs of abuse, such as cocaine, and stress regulate these covalent modifications to DNA and histones, as well as the enzymes that catalyze the reactions. And it's been widely investigated how gene expression at the mRNA and protein level changes following occupancy of single histone post-translational modifications. So what I'm trying to say is throughout the literature, we have kind of a broad sense about the concentration of these modifications and how they can affect certain cocaine-associated behaviors or cocaine-associated memories. Yet the results of this combinational effects is kind of lacking in the literature. How do they interact? When the literature was into detail about different levels of histone marks, we typically look at them in a singular fashion. And I think we might be missing a bigger piece to the puzzle by doing so. And so that's why my project aims to establish if bivalency, a novel mechanism that happens to be found in impacting cellular fate and has been implemented in cancer, is also impacted with cocaine exposure. And so bivalency is when two opposing methylation marks, particularly H3K4 trimethylation and H3K27 trimethylation, are found in the same nucleosome or in the same nucleosomic steric Hendrix space. And so what happens when you have both H3K4 trimethylation and K27 trimethylation together is you have a poised gene. And what this means is that following resolvement, meaning removal of one of these marks, you either have an activated state or a repressive state. But poised meaning that it's very flexible to be able to switch to those states. And it's been really well documented in embryonic stem cells that the resolvement of bivalency can actually impact cellular fate from creating different cell types definitively down the line. A lot of groups have started exploring bivalency, but not in a true bivalent way. And what I mean by this is bivalency is really the combinational space occupancy of these two marks together, the active and repressive marks together, whereas some people have looked at mixed cellular populations. And so this is why methodology is really important here. So if you're looking at just a normal single chip experiment, you can IP for K4 and you can IP for K27 in different mouse regions, brain regions. And you might see that, oh, we have increased level following cocaine in both. But we can't definitively say that they're in the same space. True bivalency, again, means that the nucleosome can be impacted by both the K4 and the K27, and they're within the same cell in the same stereo Hendrix space. And so just on the right side of this panel here, we're looking at examples of bivalency or not. So full co-occupancy or full bivalency would be that you have two marks that are right next to each other together. No co-occupancy means that you have some that have just one mark and some that just have the other mark. And you could also have partial, where sometimes you see like K4 by itself, and other times you see K4 with K27, but not always, and vice versa. So I think this is just really important to kind of establish bivalency versus mixed cellular population. And so I wanted to ask, does cocaine influence bivalency, specifically at NR4A1, which is a favorite gene of the lab? And so NR4A1 is an activity-dependent immediate early gene that binds DNA and recruits histone-modifying enzymes. Cocaine activates NR4A1 and impacts multiple downstream targets, such as CART-PT, THDAT, BDNF, that have been implicated in drug abuse. And we also found that NR4A1 is involved in numerous drug-related behaviors. From our own lab, we showed that NR4A1 expression impacts cocaine CPP and cocaine self-administration. It's also been found that NR4A1 expression impacts cocaine sensitization as well as amphetamine-induced locomotion. And so my hypothesis is that cocaine resolves bivalency at NR4A1. I think that perhaps NR4A1 has both the repressive and activating marks, and when cocaine comes on board into the system, it actually resolves bivalency, causing removal of K27, thus activating NR4A1 by leaving K4 on board. Alternatively, I could be wrong, so I have a second hypothesis that cocaine actually induces bivalency at NR4A1, then maybe we started out with an off state of NR4A1 with K27 trimethylation. And then once cocaine comes on board, it also draws in H3K4 trimethylation, making it into a poised state where it could be activated. In either scenario here, NR4A1 is going to be in a more active state following cocaine. A lot of publications have suggested this is true. And CART-PT expression should be increased because it's a downstream gene of NR4A1 that is activated in parallel. And so to see if either my hypotheses were true, I followed a protocol that was also used by Marco Carpenter, a recent pharmacology graduate of the lab, from his Nature Methods paper, or Nature Communications paper, 2020, where I injected mice with cocaine for 10 days, and then 24 hours after the last injection, I collected the striatum of the mouse, as you can see here on the right. I then subjected the tissue to our 3 protocol to be able to obtain transcriptomic and epigenomic profiles within the same sample. So in 2018, in the lab of postdocs, Jun Zhu, who's no longer in the lab with Liz, posted a new method that the lab has utilized greatly, where we're able to isolate the nuclear component of the neuron and then separate out the cytosolic components to be able to obtain RNA and DNA from the same sample, alternative to a trizol method. So when you do trizol methods to try and do it within samples, sometimes the DNA doesn't hold up as well in chip. But we've used the 3 protocol very successfully. And so again, we're able to, what would usually be 2 samples, to run both an epigenetic and transcriptomic profile into 1 sample. And we can directly see if there's a relationship between the RNA profile and the DNA profile within the same sample. So after separating out the RNA and DNA, I was able to subject the nuclear portion to sequential chip. So it's very similar to a single chip experiment, except there's 2 IPs instead of 1. And so essentially we'll homogenize the tissue, cross-link, sonicate, and then submit to an IP for an antibody. In this example here on the right, you can see that we're IPing for H3K4. Following the first IP, we'll submit to another IP, now for H3K27, and we'll be able to isolate bivalent chromatin. And our results show that following cocaine in the male striatum, that we have an increase of H3K27 in NR4A1, and there's partial co-occupancy as well, which was really exciting. So first and foremost, though, let me say on the top left here, we're looking at the gene expression profile, and we don't see any changes in gene expression, which was a little confusing since the literature does suggest that cocaine activates NR4A1. I will come back to why this may be in a moment. And then on the right here, we're looking at our histone post-translational modification profile at NR4A1. And so we see that there seems to be a slight increase, but not a significant increase at H3K4. There's a significant increase of H3K27, and we have a significant increase of both bivalency analyses of K4/K27 and 27/K4. And what you're looking at the bottom is that I conducted the protocol where at first I used the K4 antibody and then the K27, and I also did it vice versa with the K27 antibody and the K4 all in the same samples. So each dot you're looking at here is 1 mouse striatum, and the RNA and DNA profiles are from the same animal, just so we're all on the same page. But what's really exciting, I think, here is if we actually cover up the bottom of the right-sided screen, you might say, oh, there's the increase in K27, it's repressive. But what we're actually seeing here is a larger piece of the puzzle at the bottom where 27 came on board and it actually just made the gene potentially more poised instead of repressed. And so we might actually have a different state of the gene that we were missing before by looking at just a single chip. We also then looked at CART-PT as well, and we did not find a significant difference at CART-PT in K4 or K27, although we do have, again, these trending increases. But importantly, as you can see here, it is a little difficult to capture effects of individual variability across the groups because you can see that there is a lot of different expression values here. And so to understand the population effects, this requires correlational analysis. And so we decided to utilize Pearson's correlation to better understand our data. And so for those of you who aren't super into stats, Pearson's correlation coefficient varies from -1 to 1, and it provides us information with the strength and direction of linear relationship between continuous variables. So you're able to see if like X increases, Y decreases, if there's no relationship, or if it's as X increases, Y increases. Now, preface with sequential chip, we do violate some of the rules of independence here since we're probing for one antibody and then comparing a single to a sequential. So everything I'm about to show, I'm not going to get into much detail because we will consult a statistician on proper analysis here. I'm not an expert on correlations, but I definitely think it was worth probing into the data more to get like a populational effect. And so what we find is that cocaine causes correlational changes between the transcriptomic and epigenomic profiles. So on the left here, we're looking at the male striatum vehicle, which is our saline group, and then on the right, we're looking at our male stratum cocaine. And don't worry about the X and Y axes here. The main thing I want us to focus on are the boxes that are outlined in black. And so what we're looking at here is these axes are the same. It doesn't really matter what they are for the moment. But on the left with the saline, we're seeing some boxes in blue and in lighter yellow, which indicates either no relationship or a negative relationship. And then when we add cocaine, we're actually seeing a large flip to a positive correlation between things that were once negative. However, this is definitely selective, as you can see at the top here in both boxes, that we have high correlations that match sometimes. So it definitely looks like we're having an effect of cocaine between these correlational relationships. And our lab is working, again, to consult a statistician to kind of dig in deeper to analyze these correlations further. So the next step, besides continuing to do the correlational analysis, is I'm hoping to run intact into cut and run, or Icarus, as we like to call it, to kind of, one, confirm the sequential chip findings, but also to be able to dig even deeper into a cell type specific analysis of bivalency. And so Icarus, as we like to call it, is a combination of two methods you may have heard of: isolation of nuclei tagged in specific cell types, and then cleavage under targets and release using nuclease. And so our lab has generated double transgenic mice that have an affinity tag in either DRD1 or A2A containing medium spiny neurons. We're then able to use an IP-based method to isolate these specific cellular populations, and then take those populations and run them through cut and run, which will allow us to understand the DNA protein interactions through sequencing. And so it's essentially similar to a ChIP-seq, but it's a lot cheaper and more efficient. So that's my work in progress at the moment, and I just wanted to take a second to thank everyone that's been supporting me and helping with this project. So thank you to the Heller Lab, particularly Liz, for the mentorship and guidance. Marco, as I mentioned, has been helping me a lot as well. Shuo, one of our postdocs, Keegan, another graduate student within the lab, and of course Chloe, the undergrad I mentioned at the beginning of my talk. I'm going to say thank you to everyone else in the lab as well as our alums, my thesis committee, and of course my funding, the Addiction T32. I'm really thankful to be a part of this grant, and it's really helped propel me forward, especially with, as Julie mentioned, the recent second author neuron publication that just came out. So thank you all for your time today. Thank you for T32 funding, and I will gladly take any questions.
Great, thank you, Delaney. If you have a question, you can either put it in the chat or just unmute yourself and go ahead and ask. I may quickly start off since I'm already talking. So given the fact that genes, a gene of interest might be expressed in say, a GABA cell and a glutamate cell, why is this idea that the repressive or activating marks would need to happen in the same cell? I mean, we have this sort of tissue-specific, and activity-dependent gene regulation all the time happening in all kinds of cells. So this idea of it necessarily having to be in the same cell, I mean, why do you think that's necessary, number one? Because you could still get this, bivalency and this, downstream physiological significance of the gene being turned on or turned off under different conditions. I'm just curious your thoughts on that.
Yeah, I think a lot of it has to do with, this might sound cheesy, but understanding mechanisms in specific cell types. So I hear what you're saying, but I think if we're, particularly my interest with the medium spiny neurons, if we see that the bivalency is being driven by D1 and then A2A containing medium spiny neurons are completely not bivalent and they're just off all the time, it might create different interventions for these specific cell types or different ways to edit down the line. So my end goal is really to use the intact to cut and run because we're able to look at the specific cell types. So I hope to use, again, just heterogeneous striatal tissue at first to kind of confirm my sequential chip findings with cut and run, and then be able to dig into that cell type specific manner. And then actually down the line hopefully do some CRISPR editing within specific cell types to do cocaine-associated behaviors like cocaine CPP. Um, but I think really understanding that if that cocaine on board is affecting a specific cell type with bivalency could be really useful down the line to understanding those downstream mechanisms or creating some sort of intervention for cocaine associated behavior. But I'm definitely trying to start with like the molecular components first before I jump into that behavioral assay area.
Right, makes sense. Any other questions from the audience? No? Okay, sorry, let me, oh, there, I'm looking at the chat now. Okay. Well, if there aren't any, I guess we're a few minutes early, but we can move on to our next speaker. And our next speaker is Daniel Palamas. I'm sorry, Daniel, if I mispronounced your last name. Daniel is a graduate student in the lab of John Danny, and he comes from us fairly locally from Swarthmore College out in Swarthmore, and spent some time with a former NGG professor over at Temple, Lynn Kirby. Daniel has been very active, as all of our pre-doc students have been, and has also presented again virtually at a conference. And now I can't, there's too many things on my little screen to show up which conference you were presenting at. Sorry, Daniel. But he's been very active, and he's going to tell us today about his work on sex differences in opioid withdrawal behavior and physiology. Actually, no, let me finish my nice introduction. He's also very active in sort of outreach and GLIA. I mean, I have to say our students are really using this training period to not only develop their research skills, but also their own individual mentoring and outreach and teaching skills as well. So anyway, back to science, and Daniel's talk is on sex differences in opioid withdrawal behavior and physiology.
Thank you so much, Julie. That was very kind. And even before I start, I'd like to say thanks to the training grant. This program has been super helpful, especially the journal club has been great for understanding the field in general, and especially the clinical side, because coming at it only from the preclinical side, I find it just extremely helpful to get the perspective from everyone in this group. So yeah, I'm looking at sex differences in opioid withdrawal, specifically behavior and VTA physiology. And this being a progress report, the sex difference effects have all come within the last year, and I'm super excited about it. Just as a general outline, I'm going to give some background on why I'm performing these studies and recap what I've shown in the past. And then the bulk of the presentation will be considering sex as a biological variable, which gets overlooked far too often, especially in the VTA, as I'll talk about. And then finally, I'll discuss some tests of negative affect and ultimately trying to reverse the negative affect pharmacologically. So why study negative affect during protracted withdrawal? Well, George Koob is very into this idea. He deems it hyperkatifia. That's a phrase that he came up with himself, but it is composed of negative affect, dysphoria, anhedonia, anxiety, and all these things in combination contribute to relapse. And another thing that doesn't get talked about nearly enough is that people that misuse opioids are 50% more likely to have suicidal thoughts. And that is even accounting for comorbidity of clinical depression, which is also increased in people with opioid use disorder. So the area of interest for me and much of the Danny lab is the ventral tegmental area. And importantly, this is central to the euphoric effects of opioids. Also, people have shown that during withdrawal, GABA release probability is increased. And more recently, people have also shown that VTA GABA is aversive. These were optogenetic studies and found that increase in GABA tone in the VTA caused aversion in rodents. So the VTA is clearly very well poised to play a role in negative affect, but people haven't really tested this so far. The first question, though, I had before I even got to studying any behavior was, does chronic morphine impact VTA plasticity? And this is what I've showed previously. I recorded from dopamine neurons in the VTA and isolated GABAergic input. After recording it for a baseline period, I induced LTP with a protocol developed by the Cower lab. And you can see that the white circles, which were the saline-treated control animals, they did have long-term potentiation of these inhibitory inputs, whereas the morphine-treated animals did not. So the takeaway here is that morphine impairs inhibitory plasticity in the VTA, and I'll talk about the presumed relevance for that in a moment. One final piece to the story I wanted to add is the what we know and don't know about sex differences as it relates to opioid addiction. So in human patients, males are more likely to abuse opioids, but females telescope faster, so they develop addiction more quickly, and they report more negative affect. And there have been several things shown related to sex differences in rodents. However, if you look on the right side, this is a table that I quickly compiled of the sort of the timeline of relevant VTA studies, including from our lab. And disappointingly, they all either don't reference females or use exclusively males, except for one study, which did not note any differences. So right away, I knew I had to do at least due diligence to include males and females. And when I separated out the males and females, you can see in C and D, I noticed that the females seem to have some intermediate form of LTP. They weren't knocked down to the same degree as the males. So I found this quite surprising and exciting. And then I wanted to go back and look at baseline inhibitory activity rather than induced plasticity. So in figure A on the left, I'm looking at the paired pulse ratio, which is calculated as the ratio of the second peak. This is an electrically evoked peak, the ratio of the second to the first. And you can see that in male treated animals, this is the gray on the left, morphine reduces the paired pulse ratio. And we take that to mean that there is an increased release probability of GABA onto dopamine neurons. However, this effect does not occur in females. And this effect is recapitulated when looking at spontaneous iPSC frequency, which is also an indicator of presynaptic release probability. So this is a difficult graph to understand. On the y-axis is the cumulative probability, and on the x-axis is interspike interval. So you can reference the example traces on the right if you need to. But in short, the morphine-treated male mice have more activity, a higher frequency, and that's shown as a decreased interspike interval. And interestingly, looking at figure C, which is the iPSC amplitude, both males and females have an increase, and that indicates that there's a convergent change in postsynaptic activity, something like transport of GABA receptors to the synapse. But ultimately, my takeaway here is that male mice only have this presynaptic increased probability of release, and that in turn may cause the LTP GABA occlusion that I observed earlier. So finally, this gets to the crux of my question, which is, does pharmacological intervention targeting inhibitory plasticity mechanisms normalize negative states during withdrawal from chronic morphine? And first, to get at that question, we have to look at the mechanism of LTP GABA. So after high-frequency stimulation, the Cowra lab showed that nitric oxide synthase in the postsynapse creates nitric oxide, which is diffused to the presynapse, and via a signal transduction pathway leads to increased GABA release. So I figured that an L-name drug, which is a nitric oxide synthase inhibitor, given during withdrawal, might reverse some of the effects that I'm looking at. So this is the timeline. Mice were treated for 5 days with twice daily injections of morphine. Then they underwent spontaneous withdrawal for a full week. On day 6, so this is 1 day after morphine withdrawal begins, I tested for spontaneous withdrawal signs. And then a week later, I tested for a negative affect using the open field test, splash test, and tail suspension test. And during that 7-day withdrawal period, mice were drinking on average 100 milligrams per kilogram per day, which sounds high, but is exactly in the therapeutic range that people have been looking at in the past. So first, looking at spontaneous withdrawal signs, you can see in A, looking at total withdrawal signs, B, wet dog shakes, and C, jumps, which are classical signs of opioid withdrawal, that in every scenario, morphine increases withdrawal signs and L-name reduces the withdrawal signs. And I didn't share this before, but that has been shown in the past and was another reason why I thought L-name could be a great drug to consider when it came to looking at negative affect. So now moving on to the withdrawal day 7 results. So this is an open field test used mostly as a control, but could also capture some changes in anxiety in theory. I didn't see any changes in anxiety which would have appeared as thigmotaxis or preference to avoid an inner zone. And it also measures locomotion. This will capture if an animal has some physical impairment. And I did actually notice that the females treated with morphine and L-name had some reduced distance traveled. So this hypolocomotion was something that I had to consider, but I think ultimately is not going to explain any of the affective results. So the first main affect test that I wanted to look at was the tail suspension test. And this is very similar to the forced swim test, if you're familiar with that. And mice are suspended by their tail, and it's a measure of behavioral despair. So animals will initially squirm and appear very uncomfortable, but eventually will give up and will become immobile. And so one would predict that morphine-treated mice give up more easily. And my prediction here was that L-name would return that latency to the first immobility. And that is exactly what I saw in the males. And then very interestingly, the females, L-name did increase the latency. However, there's no difference between the saline and morphine-treated animals. So that could have arisen because of several different reasons. First, the saline injections alone could have caused some stress in the females. And whereas people haven't looked at sex very much for this opioid withdrawal effect, it has been studied extensively with regard to stress and depression-like behaviors, and females can be more susceptible to stress. So I'm currently actually testing a group that has no injection at all, and it would be interesting if that result was up higher, closer to 100 seconds. Another possibility is that the tail suspension test simply doesn't measure the affective changes that occur in females. This behavioral despair could be different than other affective things. So I wanted to look at the splash test, and that is essentially a measure of self-care. So the mice receive a sucrose spray, 10%, onto their back, and that causes grooming behavior. And surprisingly here in the males, I see no difference. But in the females, I saw exactly what was seen in the males on the tail suspension test, wherein the morphine-treated females have less grooming and LNAM returns that effect. So in summary, chronic opioids impair LTP, and that also has differential impacts on sex when it comes to physiology and behavior, and the nitric oxide system may be a target for opioid use disorder. And the final thing I need to do is determine whether the VTA is truly causing this effect or simply a byproduct, which would be a within versus outside systems effect. And finally, I'd like to thank the lab and thank the DeBiase lab, who also has helped immensely. I'd like to thank my thesis committee and the funding sources. Thank you very much.
Great. Thanks, Daniel. Questions from the audience? Gotta be some questions.
I thought that was intriguing, and I was interested. Obviously, you chose this path in terms of LNAM for your intervention. Any thoughts about something that you might do therapeutically, pharmacologically, that would go at the lack of LTP in the VTA, perhaps by doing, I don't know, GABA B agonism, either directly with the GABA B agonist or with a positive allosteric modulator? Any thoughts about other candidates in addition to the path that you're following?
Yeah, that would be super interesting. So one of the reasons that I chose LNAM is because it is independent from the sort of initial cyclic AMP mediated changes that are caused by opioids. But yeah, something targeting GABA B could be super interesting. And another thing that I've definitely considered that the lab has looked at before is chloride extrusion in GABA neurons. So the lab has shown that within the VTA, there are local interneurons, and their chloride homeostasis has been altered, and that causes them rather to inhibit VTA dopamine neurons to be neutral or even excite the dopamine neurons when the GABA neurons are activated. So we actually have a drug that can reverse that effect, and that could be in combination to the nitric oxide effects or separate. So
the reason it came to mind is that in addition to the fact that we've been fiddling around with GABA B agonists, and of course baclofan's around, not a really great therapeutic agent because of its short half-life, but finally people are doing the GABA B PAMs. But it would just be, it's interesting to know that there were some clinical publications that baclofen reduced pharmacologic opioid withdrawal, and it was one of the things that they were using to attempt to treat it at one point, a non-opioid way of going about it. So interesting to think about more than one way to float down the stream.
Yeah, certainly. I would love to have a little barrage of pharmacology effects. I'll definitely look into that.
I had a quick question. If you looked at all into estrus cycle measurements and how that affected your outcomes.
I have not. Yeah, I would love to do that. At the very least, I'm hoping to overreactimize some mice and see if I can, if the effect goes away, and then return that with some hormone initiation. But yeah, that's something that I think that would definitely add to the story. And yeah, that's one of my number one priorities, definitely.
Um, Actually, related to sort of those sex differences, has your lab or anyone looked at this idea that, you know, the increased release of GABA on dopamine happens in males but not females? Has anyone actually looked at subsequent dopamine release after chronic morphine in males versus females? Is there a difference there?
Oh, that's a good question. So measuring in the accumbens, you mean?
Right,
Yeah,
yes.
I don't know if people have tested that. And yeah, that's certainly the ultimately relevant thing that we'd like to find out. And another thing that I'm really curious about, so um, there is some differentiation between medial, lateral, VTA, and substantia nigra. So I'm really curious if there's something like a slight change in the male and female location of this effect. Um, that's kind of what the Lamel lab has been looking at when they study aversion. There are some big differences in medial versus lateral VTA. So it could be that this is something of a little pocket, and that's why it's male only. And for example, if females had progressed to a later state in the um, addiction-related behaviors, that the physiological effect has already sort of moved on from the VTA or something like that.
Interesting. Yeah, good point. And it would be interesting to see how that maps onto these two behavioral differences that on first, you know, the TST versus the sucrose splash, because
Yeah.
while sucrose splash, of course, it's grooming, but it's also rewarding to lick the sucrose off of your, you know, coat.
Right. That is a
You
complication
know, it is kind of
for sure.
interesting to see how they might be, if there's any relationship there. But
Definitely.
cool,
Yeah.
very cool. Any other questions?
I have a couple for you, Dan. First,
Sure.
how do you plan to see whether this nitric oxide effect is VTA specific? Can you inject L-name with a cannula locally, or is there some like genetic way to knock out nitric oxide or something like that?
Yeah, you can inject it locally. The thing that I'm doing right now, of course, is I want to test the VTA effects with LNAME. But yeah, you can inject it locally, which would be the simplest. And then also something that I'm going to consider is um, optogenetic alteration. So sort of like reversing the plasticity optogenetically. But yeah, LNAME, just an intra-VTA injection is the most straightforward way.
And Liz had a very important comment for you. If you are going to look at hormone flux, you should do it in both males and females, as both sexes exhibit variable levels. That's true of sex hormones across time.
Great. Yeah, appreciate that.
Cool. Okay. Thank you, Daniel. I guess we can move on to, we're running a few minutes ahead of time, but that's okay. To our next speaker, who just asked a question, Darryl Eckert. Darryl is a thesis student in my laboratory. He comes to us from Emory University and also spent some time, you know, I have to say that most of our pre-doc students spend some time as in research labs as technicians prior to joining the, in fact, he's also a neuroscience graduate group. And I think that really does help with sort of the movement and the productivity. So Darryl was over with Seema Bhatnagar over at CHOP and then joined the neuroscience graduate group. He has, since the last report, has a paper in review of behavioral brain research in a slightly different area, looking at AMPK, metabolomics, gene and serotonin response. But his talk today is going to be on the bidirectional relationship between opioids and disrupted sleep. Take it away, Darryl.
All right, thanks, Julie. Can everyone see in here? Yeah, so for my thesis project, I've been interested in the interaction between sleep and opioids and how they affect each other. One reason this is important is because while we all know that opioids are effective at managing pain, they also carry the risk of being very addictive. And in doing so, it can be hard to remain abstinent from opioids. And when one begins to be abstinent without opioids, there can be pretty intense withdrawal symptoms. The ones I want to focus on are the sleep disturbances. So these happen acutely, but can also be kind of lasting and psychological. And these negative withdrawal symptoms, especially and including disrupted sleep, can make it really hard to remain abstinent from opioids. So my idea for this project is kind of, if we can understand and treat this, maybe the withdrawal symptoms won't be so bad and it can be easier to remain abstinent. And again, this is important because even without opioids, poor sleep just by itself increases one's sensitivity to pain and impulsivity. It impairs memory, creates stress and negative affect. And so one can imagine that all of these together create a greater risk for relapse. And this also creates kind of a vicious cycle between disrupted sleep and opioids. So on the left, this shows people that are dependent on prescription opioids. They have a higher subjective score on the Pittsburgh Sleep Quality Index and the Insomnia Severity Index, just indicating that overall they get worse quality sleep than controls. On the right, you have a survey from 60,000 adolescents showing that those that sleep less than 6 hours have a significantly higher odds ratio for tobacco, alcohol, marijuana, and other drugs of abuse, including opioids. So there's kind of this feedback cycle that opioids disrupt your sleep, and then disrupted sleep can increase your affinity for drug use. And so that we want to see what's going on in the brain and how can we kind of stop this vicious cycle from continuing. So the way we did this is looking at a chronic morphine paradigm in mice. We did a oral forced morphine paradigm, so we dissolved morphine in saccharin to mask the bitter taste. We did this because there's no stress of injections or invasiveness of a surgery with a mini pump, and it's really easy to swap out the morphine in the drinking water so that you can make the mice go through withdrawal. In collaboration with our colleagues at the Sleep Center right down the hall, we did electroencephalography at different time points of this morphine paradigm. So we would get 24-hour periods of sleep analysis at baseline, at acute morphine, at chronic morphine, and at acute and also protracted withdrawal. And so what we see here, first during the lights off time, so this is when mice are typically more active, we see that on acute morphine as well as chronic morphine, these mice spend even more time awake during the active period. So this is kind of an exacerbated circadian rhythm. They're more awake during the time period when they are typically awake. And then into the first 24 hours of withdrawal, we actually see morphine withdrawal effects during the lights on or the inactive period. So during the first 24 hours of withdrawal, mice are more awake when they should be asleep. So kind of the summary for this is that acute and chronic morphine kind of exacerbates a normal circadian rhythm, and then the first 24 hours of withdrawal kind of flips that circadian rhythm, and by 2 weeks of withdrawal, it returns to about baseline. And then on the right, you can just see kind of an hour-by-hour graph how during the chronic morphine, they have this really elevated weight phenotype. Same morphine paradigm also induces affective behavior changes 2 weeks later. So Dan has kind of explained some of these already. I didn't see changes in the open field, but during 2 weeks of withdrawal, we see decreases in grooming in the sucrose splash test and significant increases in the latency to groom. So this indicates kind of a decrease in self-care. And we also see differences in the tail suspension test 2 weeks later. We also followed this up and looked at the brains of these mice. So we looked in the cortex and did qPCR for a number of markers that have been shown to be increased in sleep disruption in general without opioids. So we have TNF-alpha, which is a pro-inflammatory cytokine. IBA1 is a calcium-binding protein that is used as a marker of activated microglia. Nitric oxide synthase, which Dan already alluded to, cyclooxygenase 2, an enzyme involved in forming prostaglandins and is also the target of NSAIDs and anti-inflammatory drugs, and then amyloid precursor protein. So this is the precursor molecule that generates beta amyloid and is kind of used as a neurodegenerative marker. So the overall trend is kind of on chronic morphine, a general increase in these neuroinflammatory and metabolic and degenerative markers. So basically the summary so far is our chronic morphine paradigm exacerbates sleep disturbances both during opioids and during withdrawal, has behavioral consequences 2 weeks later, and then some neuroinflammatory consequences in the cortex too. But next we kind of wanted to drill down a little more specifically where in the brain these sleep effects and behavior effects are coming from. So we did a screen of the immediate early gene CFOS, which we probed with immunohistochemistry to use as a marker of activated neurons during acute drinking of morphine or chronic drinking of morphine, both with a stimulus injection just to robustly enough detect the CFOS markers. And we looked across a number of brain regions that have been implicated in reward and/or sleep. And what we found is 2 significant increases in acute morphine, the lateral hypothalamus and the paraventricular nucleus of the thalamus. And within those, the lateral hypothalamus kind of returned to baseline in the chronic drug state, but the paraventricular nucleus of the thalamus didn't decrease, so it kind of remained active. And so this was interesting to us as a brain region of interest, potentially at the nexus of opioids and sleep-wake. First through our CFOS data, which showed increased and sustained neural activity in response to acute and chronic morphine. And then we also thought that this is consistent with this increase in wakefulness, increase in morphine-induced wakefulness that we see at the acute and chronic time points. And furthermore, other groups have shown that the paraventricular nucleus of the thalamus controls wakefulness, so optogenetically activating PVT neurons will wake an animal from sleeping, and then optogenetic or chemogenetic inhibition will decrease the latency to sleep. So activating these neurons wakes an animal up, and inhibiting these neurons puts an animal to sleep. These neurons also control the withdrawal behavior from opioids and specifically their projections to the nucleus accumbens. And then finally, these paraventricular thalamic neurons respond differently to morphine. So if morphine is injected in the light cycle versus the dark cycle, these neurons have different firing properties. So our data as well as literature kind of points us to the paraventricular nucleus of the thalamus as a really interesting region for the connection of opioids and wakefulness. So our hypothesis for this is that the sustained neural activity in the PVT that we see is responsible for the chronic morphine-induced wakefulness, and we plan to test this using DREADs or designer receptors exclusively activated for by designer drugs. So with a virus or sometimes genetically, you can introduce this engineered receptor that no longer responds to acetylcholine. It's an engineered muscarinic receptor, and instead of responding to acetylcholine, it now responds to a normally inert ligand called CNO, or clozapine N-oxide, or a number of other drugs that do the same thing. The idea being that you can inject CNO, or something that normally does nothing to an animal, but with these specially engineered receptors, you can specifically activate or inhibit these neurons. So I'm injecting this Cre-dependent DREADS virus into the PVT. So it's contained within a double-floxed inverted open reading frame, and only when it comes into contact with the enzyme Cre recombinase to take out the LOX-P sites and orient it correctly will this inhibitory DREADS virus become functional. So I injected this virus encoding the inhibitory DREADS virus into the PVT of a more Cre mice. So we have mice that express Cre recombinase only in mu-opioid receptor expressing cells, and showing up on the internuclear membrane here. So what we see here is green showing mu-opioid receptor expression in the PVT, and red after 4 weeks, this virus with an M-cherry reporter is shown here in the same mu-opioid receptor expressing cells. So this is going to allow us to inhibit specifically mu-opioid receptor expressing neurons within the paraventricular nucleus of the thalamus, and our hypothesis is that inhibiting these mu-opioid receptor expressing neurons, which we saw are highly active during the high wakefulness period, will decrease this morphine-induced wakefulness. So our summary so far is that we have this relationship between morphine and sleep disturbances with behavioral and neuroinflammatory consequences. We've shown that opioids disrupt sleep. We've shown opioids alter behavior. Potentially, paraventricular nucleus of the thalamus mu-opioid receptors are responsible for this disrupted sleep. And that's coming in the future directions soon. But next, I kind of wanted to go to the other side of the vicious cycle that I talked about earlier and see whether disrupted sleep can affect opioid use. So this is important because 30% of opioid users cite sleep disturbance as a reason for relapse, and we wanted to kind of disentangle this direction of whether disrupted sleep can increase the potential for opioid use and abuse. The way we're testing this is with a chronic sleep deprivation paradigm. So again, in collaboration with our colleagues at the Sleep Center, we have this chronic sleep deprivation paradigm where we will deprive the mice for the first 8 hours of the light cycle, 3 days a week. This gets repeated 3 times, so 4 total cycles of a week of sleep deprivation. And following this, we did a 2-bottle choice experiment. So we can compare controls versus chronic sleep deprivation at 4 weeks and say, between saccharin and morphine, do you prefer saccharin or do they prefer morphine? And the way we did this sleep deprivation experiment, there are a couple of ways to sleep deprive mice, and unfortunately many of them come with confounds, including stress or loud noises, or investigator labor-intensive experiments. So the couple that are kind of most popular are the gentle handling method, where an investigator just kind of watches the mice and pokes them or moves them or uses a paintbrush to make sure that they stay awake whenever they appear to fall asleep. The cons to this is that it's very labor-intensive. It can be stressful, and there can also be kind of microsleeps when you realize that they're falling asleep before you've intervened. There's also an extended wakefulness paradigm. So this involves just kind of placing novel objects in the cage every half an hour or so, and this novelty by nature keeps the mice awake. And so they just don't fall asleep because they're intrigued by the novel objects here. So we tested both of these out and a radioimmunoassay for corticosterone shows that the gentle handling method significantly increased CORT levels in the blood, while the extended wakefulness was exactly the same as baseline. So what we ended up going with for this extended sleep deprivation paradigm is using the extended wakefulness method of sleep deprivation. We also used a couple automated systems to track both the morphine preference and their sleep disruption. So we used infrared sensors on the top of these cages that kind of track activity non-invasively, continuously, and we can convert that to a sleep score because 40 seconds of no activity correlates with electroencephalography measures of sleep in the system. And then we also have these automated lichometers that have a small computer chip here and infrared sensors under these two tubes that can tell when and what tube the mouse is drinking from. So we kind of put these together to monitor the sleep deprivation effects on morphine intake. So this slide just shows the sleep deprivation data. We did see a slight decrease in body weight gain between the controls and the sleep deprivation group, but both had a healthy body weight gain over the course of the experiment. On top here is minutes asleep. So minutes asleep every 12 hours. So during the sleep deprivation in the lights on period when we deprive the mice, they have significantly less sleep. And interestingly, the recovery sleep doesn't happen in the light cycle, which is typically when mice are asleep. The recovery sleep happens at the immediate next dark cycle. So as soon as these mice are sleep deprived, the next dark cycle, they sleep more than controls. And this actually increases over time. So you can see this kind of increasing burden of sleep debt, if you will, where the second week of sleep deprivation has increased recovery sleep from the first, and the third has even more so. So by week 1, we're getting about an hour of recovery sleep at the next dark period. By week 2, it's about 2 hours, and by week 3, it's about 3 hours of recovery sleep. So this sleep deprivation is really impactful, and the mice show it week to week. And so immediately after the last sleep deprivation, we put mice into the morphine 2-bottle choice experiment to test whether they would have an increased preference for morphine, which was our hypothesis. We thought these mice that were sleep deprived would have an increased preference for morphine. And what we ended up seeing was actually the exact opposite of our hypothesis. So our control mice in blue developed a preference for morphine that topped out at about 75 to 80%, whereas our sleep deprivation mice had no preference. They were essentially right at chance, or 50% the whole experiment. Their drinking behavior was the same, so they ingested the same amount of liquid. It was just a preference for the control group and no preference for the sleep deprivation group. So this was really interesting to us based on human reports and clinical data. This is kind of the opposite of what we would expect. So we thought, is morphine no longer rewarding, or do mice need less morphine for it to still be rewarding? I think both hypotheses are possible based on this data. So we followed this up. This is happening now. What we're doing is the same sleep deprivation paradigm, and we're going to repeat instead with conditioned place preference instead of 2-bottle choice. So this will tell us whether the mice have a rewarding association with morphine. So if we don't see a rewarding association in the CPP, we would interpret it that morphine is no longer rewarding. But if both groups have a preference for morphine, or the sleep deprivation group has even more of a preference for morphine, we would conclude morphine still is rewarding, and maybe it's a dose type effect. The sleep deprivation could change the dose at which morphine is rewarding. We also took advantage of these automated lichometers to get more detailed data. So this is a continuous expression of the two-bottle choice. So again, instead of the previous data that I showed, which was hand-scored once a day, this is kind of a continuous preference that also showed that the orange sleep deprivation group doesn't show a preference, but the blue control group does show a preference for morphine. This also kind of was able to average when each group was drinking most of the morphine. So this is over the 7-day two-bottle choice paradigm. It shows an average of the light cycle drinking behavior and the dark cycle drinking behavior. And what we see is the increase in preference that we see in the control group looks to happen at the end of the dark cycle into the early hours of the light cycle. So interestingly, looks like a circadian component to the morphine drinking preference. It's not kind of a stable increase in drinks throughout the light and the dark cycle.
So 1 minute warning, Darryl. Since I'm the moderator, I don't want to give you any extra time.
Great. So we also looked in the brain at IBA1, a marker of activated microglia that we had seen earlier in the sleep study. And what we found is that sleep deprivation increases IBA1 expression, and this looks to be blocked by morphine. So there appears to be this neuroinflammatory component to sleep deprivation influencing opioids. And so in summary, we've shown that opioids can disrupt sleep and alter behavior, potentially driven by paraventricular thalamus mu opioid receptors. Disrupted sleep can alter opioid intake, not in the way that we thought, but we're still following up on this. And so with the future directions, we kind of aim to fill in this whole cycle here. Our future directions is do a sleep study with DREAD's inhibition of the paraventricular thalamus mu opioid receptors to block the morphine-induced wakefulness. You can also follow up by looking at the inputs and outputs of these neurons. We're going to sequence these mu opioid receptor expressing neurons in the paraventricular thalamus to get a profile of what exactly these neurons are, and we'll finish up the conditioned place preference with sleep deprivation. So thanks to everyone. My acknowledgements page is rapidly expanding over my time on this training grant as a lot of people contribute to this project, and I'm very thankful for it. So thanks, everyone.
So we're about a minute over. I don't know if we want to have time for questions or if we should move to the next speaker.
Just wanted to say it's very intriguing, Darryl, and I think that's the glory of having an elegant animal model is that you can begin to try to parse some of these things, because even though we do have human reports about sleep deprivation and sleep difficulty leading to taking of opioids or alcohol or other things to get back to sleep, it really needs to be parsed as you are. So congratulations on parsing it.
There aren't any burning questions. You all know where Darryl lives, so we can certainly reach out. I think we'll go ahead and move on to our next pre-doc speaker, Rachel Herman, or Ray. And like our other phenomenal students, Ray comes from a semi-locally and actually is a fellow TERP, which I didn't realize for some reason. She got her bachelor's degree at the University of Maryland, College Park. She's already has at least one paper manuscript, at least in preparation, with a rotation, I'm guessing, with Mariella DiBiase. And she has presented on her work from Heath Schmidt's lab on the glucagon-like peptide receptor at the Society for the Study of Ingestive Behavior. She's also very involved in outreach. She's an Upward Bound lecturer and works with the Philly Brain Bee outreach and STEM Scholars lecturer. So we're excited to hear her talk today on liraglutide decreases nicotine taking and body weight gain during nicotine abstinence. Ray.
Thank you so much. All right. I just want to say I'm so excited to be a part of the training grant and hear all these great talks. So today I'll be talking about a very translationally focused project that I've been working on over about the past year, which is looking at, as you said, how liraglutide decreases nicotine seeking and prevents body weight gain during nicotine abstinence. So as I'm sure everybody is familiar, cigarette smoking continues to be the leading cause of preventable death in the United States. But one very crucial aspect of the difficulty of quitting nicotine consumption is the fact that the majority of people who quit smoking gain weight. And this can be an issue for two reasons. Firstly, because this possibility of post-cessation weight loss can be frequently a deterrent for people not wanting to quit smoking. And then secondly, for a significant minority of people, this weight gain can lead to the development of chronic illnesses like obesity or type 2 diabetes. So ideally, our treatments for nicotine use disorder would consider this issue as part of the barrier to successfully quitting nicotine use. However, the current treatments we have don't completely address this. So nicotine replacement therapy, which is things like a nicotine patch that you apply, is effective in decreasing nicotine use, but it is very variable in its effectiveness. Particularly for females, it shows less effectiveness than it does for males. And treatments like nicotine replacement therapy or bupropion are known to delay but not completely prevent this weight gain during nicotine abstinence. So we're not really tackling this other aspect that can make quitting more difficult. So my lab is interested in something that we think may be a more well-rounded treatment for nicotine use disorder that addresses both the problem of nicotine seeking and this issue of post-cessation weight gain, which is targeting the glucagon-like peptide 1 receptor system, or GLP-1 receptor system. So GLP-1 is an endogenous satiation hormone and neuropeptide. It's been known for a while to regulate homeostatic and hedonic feeding, and GLP-1 receptor agonists are actually FDA approved in treatment of obesity. So it's very well established that these treatments are effective in leading to weight loss. A little bit more recent area of study has shown that because of its effects on the mesolimbic reward system, it's also effective in regulating drug seeking for almost every drug of abuse that has been investigated. So particularly, we're interested in nicotine. And because we know that these GLP-1 receptor agonists are effective in creating weight loss, and there are preclinical studies that show that systemic GLP-1 receptor agonism is sufficient to decrease nicotine intake and nicotine reward, as measured with conditioned place preference and nicotine-evoked dopamine release. We hypothesize that these GLP-1 receptor agonists could act as this really exciting dual-purpose treatment tackling both of these components of nicotine use disorder. So today I'm talking about how I explored the potential of these treatments in our rodent model. So specifically, I wanted to know what the effect of specifically the GLP-1 receptor agonist liraglutide is on, firstly, nicotine reinstatement behavior, so not just self-administration, but this model of relapse. Then secondly, if it's effective in decreasing weight gain during nicotine abstinence. And then finally, it's very important to explore the sex differences in the efficacy of these agonists, particularly given that we see different efficacies of treatments like nicotine replacement therapy. So I have two main hypotheses moving forwards, which is firstly looking at the self-administration behavior. I predicted that liraglutide will decrease nicotine self-administration and reinstatement in male and female rats. And then secondly, for the body weight component, I predicted that repeated liraglutide will prevent hyperphagia, so excess food consumption, and the body weight gain that occurs during nicotine abstinence in male and female rats. So in order to first focus on that first hypothesis, looking more at self-administration and reinstatement behavior, we use the self-administration and reinstatement paradigm where rats, male and female rats, were trained to operantly self-administer nicotine, starting on an FR1 schedule and progressing to FR3, so 3 lever presses, they receive 1 infusion of nicotine. On the last day of self-administration, we administered an acute injection of liraglutide 1 hour before their self-administration session, so we can see the effect on this self-administration behavior. Then we continued to administer liraglutide or vehicle in the vehicle controls throughout the rest of the experiment. And the animals then went through extinction where they received saline instead of nicotine until they were below 20% of their baseline pressing and considered drug-seeking extinguished. And then finally, the animals went through a drug and cue-induced reinstatement test where they had an IP injection of nicotine and were also re-exposed to drug-associated cue-light in order to provoke a return to nicotine seeking. And ideally, we would hope to see that this liraglutide treatment throughout extinction would decrease that drug-induced reinstatement. So I'm going to start by looking at the female data. So this is the acute experiment where we just administered liraglutide immediately before their final self-administration session. And here we can see on the y-axis are the lever presses. Left side is active lever, right is inactive lever, and we want to focus on the active lever since that indicates drug-taking behavior. And you can see that liraglutide was indeed effective in decreasing nicotine self-administration in these female rats, as we predicted. Very exciting. And we see the same thing in infusions. So these animals are pressing less and receiving fewer infusions after liraglutide treatment. So when we look at reinstatement, so after they've gone through extinction and then have this drug plus cue-induced reinstatement, we see that liraglutide continues to be effective after this repeated treatment. So the female rats also show decreased lever presses in reinstatement and appear to have this lower relapse occurring after liraglutide treatment. So that's all very cool. But when we look at the male rats, we actually saw something different, which was unexpected. So for the male rats, we don't find an effect of liraglutide treatment on acute self-administration when we look at lever presses and infusions. And we also don't see an effect of repeated liraglutide treatment on this drug plus cue-induced reinstatement. So in conclusion, for the self-administration portion, we find this really interesting, unexpected, sex-specific effect where liraglutide decreases nicotine self-administration and reinstatement as we predicted, but only in female and not male rats. So moving on to the body weight portion of my hypothesis. Again, I predicted that this repeated liraglutide treatment will prevent hyperphagia and body weight gain during abstinence. So in order to model this withdrawal-induced hyperphagia and weight gain, we started out by food restricting animals to encourage acquisition of nicotine self-administration. Animals then self-administered nicotine in once daily 2-hour sessions for 5 days, and then were switched to twice daily 2-hour sessions separated by 2 hours, so sort of intermittent access paradigm. Near the end of this self-administration time, we start increasing their access to normal chow, so they're able to eat more. And then right before the end of the self-administration section, we switch them to an ad-lib high-fat diet. Then on the last day of nicotine self-administration, we start treatment with either liraglutide or vehicle for our control animals. And then finally, the animals are returned to home cage abstinence, so they're no longer going in the behavioral boxes, but we're just measuring their food intake and body weight over the course of these 10 days. And so we had our 3 groups that I'll be showing right now are nicotine self-administering animals that were treated with vehicle or liraglutide. And then I also have a group that's yoked saline animals who never received any nicotine, but just went through this yoked saline self-administration, receiving saline injections, and then were treated with liraglutide. So you can see the effect of liraglutide without the nicotine withdrawal. All right, so I only have this data so far for male rats. So just to orient you to this graph, we're measuring food intake 6 hours after liraglutide treatment over the course of abstinence. And this yellow group is the nicotine control animals who theoretically would be showing this hyperphagia. Then the orange group is the liraglutide treated nicotine animals. And then the blue group is the yoked saline controls who received liraglutide. So we can see there was a significant effect of treatment over the course of this 6-hour food intake during abstinence. And if we average each animal's food intake over the course of abstinence and compare between the groups, there's a significant difference between the nicotine animals that received a vehicle treatment and the liraglutide treated nicotine animals. So it appears that this liraglutide treatment was effective in decreasing 6-hour food intake in males. But then when we look at 24-hour food intake, we don't really see an effect. So it seems like there might be a little bit of this effect happening in the beginning, but maybe it's more transient and goes away over the course of abstinence. So it's interesting that maybe the effect of liraglutide is stronger when we're looking at the more 6-hour, but fades when we look at the 24-hour time point. But nonetheless, we do see that it continues to have an effect on body weight. So if we are looking at the change from baseline body weight, again with these three groups, there's a significant effect of time, treatment, and subject. So it appears that these nicotine plus vehicle animals have higher increase from baseline body weight compared to both of the liraglutide treated groups. Although again, maybe that does show some sort of fading away by the time we reach the end of abstinence. But there's definitely an effect of liraglutide attenuating this body weight gain. And we don't see any effect when we look at 24-hour water intake, which is good control. All right, so to summarize what we've seen today, and really interestingly, we see this sex-specific effect where liraglutide decreases nicotine self-administration and reinstatement in female but not male rats. And while we only have the data from the male rats, it looks like liraglutide was effective in attenuating food intake and body weight gain during nicotine abstinence. So I think these results are really exciting, showing the potential of liraglutide as this treatment that can target both aspects of the nicotine use disorder itself and then also addressing the cessation-associated weight gain. And particularly when we see that it's effective for female rats, when things like nicotine replacement therapy are less effective for females. I think that's really a promising result. And there are also some clinical studies that show that GLP-1 receptor agonism combined with nicotine replacement therapy may be effective in decreasing relapse to nicotine use, as well as inhibiting this post-cessation body weight gain. So I think that's one potential way this could be implemented to sort of harness the best of both worlds. So just to finish up this project, I'm plan to look at the effect of liraglutide on withdrawal-induced hyperphagia and body weight gain in the female rats, as well to add to the male group I already have. And then I also want to measure kaolin consumption during liraglutide treatment in both groups of animals, since nausea can be a common side effect of GLP-1 receptor agonists. And then eventually it could be interesting to look at the neural substrates of liraglutide's effects on withdrawal-induced feeding and try to explore if this is happening more through GLP-1 receptor agonist action on this hedonic or homeostatic sort of feeding sites of the brain. Right, so again, thank you so much to allowing me to be a part of the training grant program. I'm really excited to be here. And also want to thank everyone in the Schmidt Lab for all of their help.
Great, thanks, Ray. We have a couple of questions in the chat, one from Liz Heller and one from Alice Ely. Do you guys want to ask them or shall I read?
Go ahead and ask them if you want. Yeah, go ahead.
Yeah. Somebody said something. Sorry. Please read me. The dog is very barky right now. Okay, so Alice asks, I may have missed it. Were the animals OB prone or resistant, and what would you hypothesize the difference might be, given differences in post-cessation weight gain in humans, depending on BMI?
Yeah, good question. So these were just wild type rats with no sort of measurement of that beforehand. I just tried to like counterbalance each group. But I know in clinical populations, there's a greater likelihood that you see increased weight gain in people who already have obesity. So I think that definitely could be also interesting if we wanted to sort of, it's a little bit difficult to have this like hyperphagia phenotype, which is why we have this like food deprivation followed by high fat diet and all the nicotine exposure. So that's actually a good idea for maybe beefing up the like hyperphagia effect and seeing if we could treat it more effectively.
And then Liz's question was, what is the rat model of body, where to go? What is the rat model of body image? What's the rat proxy of female humans not avoiding weight gain? Of female humans avoiding weight gain. So
Yeah,
go ahead.
that's definitely a good question too. I think this is definitely interesting because it allows us to kind of separate that component, I assume. I can't read the rat's mind, but it seems like when we still see these differences in like the effectiveness of different treatments in females versus males, and I know sometimes people will cite body weight concern as like a reason that females have more difficulty quitting, but when we also see it in the preclinical models, I think that's definitely.
Indeed, and tobacco actually made, Virginia Slims was a type of cigarette specifically targeting women who wanted to keep their weight down. Gotta love industry. Any other questions?
I have a quick one that just out of my naivete. In other models, have these kinds of agents shown sex differences, or did you manage to pick it up in this nicotine model?
Do other
The
treatments
same agents that are working here on the same substrate. I just wondered if, I know you were using it in the nicotine model. I know why you were using it, but I just wondered if, in general, since these are being examined, is it something that shows sex differences just on its own in humans, for example, or others, or other animal models?
That's a good question. I know in our work, I think definitely just exploration of the sex differences is lacking in general in the literature. So I think that's definitely something we'll see more of in the coming years. But I'd say we haven't really seen these significant sex differences in other drugs of abuse that with the effective GLP-1 receptor agonists, because we've done it mostly in male rats and have seen an effect. So this seems possibly unique to nicotine.
That's interesting, yeah, and important too. So kudos to you for parsing it out.
Thank you.
So we're at 2:31, and I believe we are ready for and thank you. I just want to thank all of the speakers so far from the pre-doctoral portion of the T32. They were really excellent talks. But now we're moving on to our postdoctoral trainees. And Anna Rose, I guess you will introduce some of these guys?
Yes, I will definitely have the honor. And basically, our first speaker today is going to be Aladdin Riyad. And Aladdin came to work in Bob Mack's lab after having his both undergraduate and advanced degrees at the University of Central Florida. He's been with Bob Mack and worked on a number of interesting projects as a chemist in Bob's lab, working both on, I believe, possible delta tracers with the delta opioid receptor, but interestingly and importantly, trying to focus on also getting a D3 specific radio tracer. It's been a long time pursuit, not just for Aladdin, but for the field. And we're very excited to hear a summary of the sort of D3 story today. So take it away, Aladdin. Aladdin, by the way, will be finishing his 3 years on the T32 November. And he basically will be fortunately sticking around and staying on the pursuit, hot on the trail. So please go ahead, Aladdin.
All right, thank you very much, Dr. Anna Rose. Can everybody see my screen correctly? I'm going to assume
Looks
that's
good.
it. Okay,
Looks good.
perfect. So thank you again. As you stated, I'm going to be talking to you guys today about some of the factors that we found that are important for the development of a D3 selective PET radio tracer. As a brief summary of the dopamine 3 receptor, it's a pharmacotherapeutic target for neurological, psychiatric, and substance abuse disorders. It's highly expressed in the mesolimbic pathway and has implications with the reward and motivation. Postmortem studies show there's an increased expression of this D3 receptor in drug-exposed brains. And unfortunately, it has a high homology with the dopamine 2 receptor at the orthosteric binding site. So when designing compounds to bind specifically to dopamine 3 receptor, we need to design compounds that can also interact with the secondary binding site. This interaction will provide specificity for a compound. A selective radio tracer for PET imaging applications is absolutely vital right now to understand its regulation and mechanisms in a variety of things, including addiction. Here are a couple of PET tracers that are currently being used for imaging dopamine receptors. As you can see, both on the left here, you've got Falopride, which is an antagonist. Unfortunately, it has the same subnanomolar affinity for both D2 and D3 receptors. On the right, we've got PHNO, which again has that same issue. It has subnanomolar affinities for both receptors. So while both of them can be used to image dopamine receptors, they cannot be selective for the 3 over the dopamine 2 receptor. So that was our goal with this project, to try and develop a D3 selective PET tracer so that we can study that receptor in detail. Moving forward, so here are some imaging studies that were done with, let's start with Falopride. Falopride is a benzimide, and it's non-selective again. It has the same affinity for both D2 and D3 receptors. So when you use Falopride, we label both of these receptors, as you can see. It's hard to tease out the signal from these images between the D2 and D3 because, as you can see in this panel over here, most regions of the brain express both D2 and D3. The exception to that being the thalamus, where you have more D3 receptors than you do D2 receptors. FTP, as you can see here in the middle, is a more selective radioligand, much more selective radioligand. It has a higher affinity for the D3 receptor than it does the D2 receptor. However, when we look at some of the imaging results, we see that the results were variable. With some of these studies, specifically the rhesus monkey number 4, who was appropriately actually named Aladdin, it looks like an actual D3 image where you can see high amounts of uptake in the thalamus. However, with the other studies, 1, 2, and 3, the other monkeys, you do not see that this consistency. You do not see that the D3 receptors were consistently labeled. So, and again, it's important to notice that both of these have a similar affinity for the D3 receptor, despite the fact that FTP was not able to consistently label the thalamus. So, another study that was done was if you deplete the dopamine using lorazepam, you can see the thalamus clearly and consistently being labeled. So this study helped us discover that if you deplete the dopamine in the synapses, using lorazepam, you are now able to clearly and consistently label the dopamine 3 receptor. This was not necessary, however, when using Falopride. It was only necessary when using FTP. So despite the similar affinities. So we wanted to move forward and see why Falopride was able to label the dopamine 3 receptor, the thalamus, without having to deplete the synapses of endogenous dopamine as you were with using FTP. Before we get into that fully, let's look at the second PET radio tracer, PHNO. Here, if you look at the thalamus, PHNO is able to bind. And again, on the right, you see that for reference, it's the thalamus that determines is what we look at when looking at a specific D3 receptor PET tracer, because it has more D3 than it does D2. So PHNO and falopride were able to label without depleting the endogenous dopamine out of the synapses. Why? Why couldn't FDP do this? So to answer this question, we looked at some other things besides simply the affinity of the compounds for the receptor. What we did was set up a beta-arrestin functional assay. So this assay measures how well a compound can compete with endogenous dopamine for the receptor. As you can see here, falopride has a IC50 value of 1.7 nanomolars, whereas FTP has an IC50 value of 611 nanomolar, a terrible IC50. So this indicated to us that falopride was able to compete with endogenous dopamine for the dopamine 3 receptor, yet FTP was not. Because in this assay, we use cells that overexpress just the dopamine 3 receptor. So it's specifically looking at how well they can compete with dopamine at the dopamine 3 receptor. Now, when we look at PHNO, we were not able to run it in antagonist mode, of course, because it's an agonist. So we ran it in the agonist mode. We see that its EC50 for the dopamine 3 receptor is, again, 1.7 nanomolar, literally identical to falopride's IC50. Its EC50 was much better than endogenous dopamine for the dopamine 3 receptor, which is about 7 nanomolar. So that indicated to us, okay, we can use this assay to understand if a compound is able to able to compete with endogenous dopamine for the dopamine 3 receptor. So it added a second layer to our workflow. A, we have to have something that binds with high affinity to the receptor. B, we want it to be selective. And then C, now we want it to not only be high affinity, but we want it to be able to compete with endogenous dopamine in a functional assay, in live cell assay. So moving forward, we have created a series of flexiprides, molecules that have a portion of them that bind to the orthosteric site and a portion that bind to the secondary binding site, which provides that selectivity. One of these compounds that looked quite promising based off of its properties and some of the results we got from binding and our functional assays was this one right here that's highlighted, HY2-92. Ho Young synthesized this compound. We found that this compound was highly selective for the dopamine 3 receptor, had about 2 nanomolar affinity for D3 versus the 265 nanomolar for D2. But more importantly, it had that magic number, which we look at as being below 2 nanomolar IC50 in our functional beta-arrestin assay. So this compound looked like it was very promising, had everything that we needed for it to be a potential candidate moving forward into a PET tracer study. Another thing that's very important before we actually move forward in putting it to animals to label the D3, to see if it could label the D3 receptor, was to ensure that there was no off-target binding. And so to do this, we sent the compounds off to get screened in the lab of Brian Roth. And what we found was that the compound had very low affinity for off-target receptors, which is very important if you're trying to make a tracer that's selective for a single receptor. Again, as you can see here, these are some more receptors that it had very low affinity for. So basically, the compound was clean, and it labeled the D3 receptor in a selective manner. So at this point, Ho Young, he went ahead and he moved forward with radio labeling the compound. It was a straightforward process from the chemistry perspective, and we went ahead and we conducted some imaging studies. Unfortunately, as you can see from the images here, the uptake in the brain was very low. It did not get into the brain as we hoped that it would be. So despite the fact that it had high affinity for the receptor and was able to compete with endogenous dopamine for the dopamine 3 receptor. So this had us wondering, why is it? There must be something else that must be going on in order to help us understand a better way to optimize our pipeline so that we can now make a radio tracer that not only has all of the properties I described before, but is also able to get into the brain. So we sat around, we asked ourselves, what is it that we could do to optimize this pipeline? And this led us to the boiled egg plot. So now this plot's very interesting here, this boiled egg plot. What it does is it looks at two parameters. It looks at lipophilicity and it looks at polarity of a compound. So based off of this, this paper that we found puts all of the molecules in what looks like an egg. Things that are in the yolk of this boiled egg, in the yellow portion here, they have the capacity to get into the brain. Things that are white tend to just go into the intestinal tract. They get absorbed in the intestines. So we needed compounds that had the chemical properties that would land them in this yellow portion. If we look at FTP, which if you remember previously it was highly selective, yet you could only label dopamine if you deplete, you can only label the dopamine 3 receptor if you deplete endogenous dopamine from the synapses. You see that right on the edge, slightly outside of the yolk. That made sense. Meanwhile, other compounds such as phthalate, PHNO, which I talked about earlier, and raclopride, atiklopide, etc., they are within the yolk. So despite that they're not selective, but it makes sense why we were able to label both D2 and D3 using them, because they can get into the brain. Some of the other compounds that we looked at that I discussed, the HY292 earlier, the one that couldn't get into the brain but had all the ideal properties, as you can see, it's outside the yolk. So it's made sense. Everything was starting to become consistent once you look at this boiled egg plot. So moving forward, Ho Young has designed a few compounds here that are within the yolk, and so ideally should get into the brain, and we are now in the process of testing them for their affinity and their ability to compete with endogenous dopamine in the functional assays. So in conclusion, a compound, a dopamine 3 receptor specific compound is provided by its ability to bind that secondary binding site. Specificity, we determine that via radioligand binding assays so that we can find compounds that have higher affinity for the D3 versus the D2 receptor. Then after we find that, we move on to finding compounds that have the capacity to compete at this about 2 nanomolar is our magic number range with endogenous dopamine, and the compound. Furthermore, the compound must fall within the yolk of the boiled egg plot, which predicts that the compound will be able to get into the brain. And that's what we currently are working on. We have some compounds. I showed you a few of them that fall within the yolk of the plot that are currently now being tested. Furthermore, we're also continuing our in silico work to find other compounds that we can test for their affinity, for their ability to compete with endogenous dopamine, and again fall within that region of the plot that predicts their entry into the brain. I'd like to thank everybody here in the night at T32 for giving me this opportunity to learn quite a bit over my time on the grant. I'd like to thank my mentor, Robert Mack, for giving me this opportunity, teaching me so much, and for all of the different projects and everything I've learned on doing the project. I'd like to thank Ji-Yoon Lee, Ho Young Kim. Ji-Yoon did quite a bit of the beta-arrestin assay work. Ho Young Kim, he did all of the radio labeling and synthesis for the compounds that we talked about today. CJ did all the in silico work. I'd like to thank Costa for some of for doing all of the chemistry for the dopamine work for the opioid work that he's going to be presenting in a little bit. like to thank Tom Graham and our previous lab member, Sean Riley, which you all know from previous years that we worked quite a bit on this project together. And of course, the cyclotron and the entire Department of Radiology here at UPenn. And with that, thank you very much, and I will take any questions if there are any.
Thanks so much, Aladdin. That was really fun to get that overview and to sort of get a sense of where things are. And it looks like you've got some hot candidates. By the way, I should mention to everybody, I think you have a K application under review, so we should be crossing our fingers for you as well, Aladdin.
I do. Fingers crossed. Fingers crossed. I'll find out very soon now, at the end of the week.
Yeah, right, in October. So we'll
Yep.
keep our fingers crossed. But in the meantime, I have a mundane question, just in terms of workflow. Since it's sort of painful to get to the point of the boiled egg and then find out that what you've discovered has all the right properties except it can't get in the brain. Is it naive to say, would you check on compounds very early for their brain penetration before doing all the other elegant work?
Absolutely. So once we find some compounds that we want to go ahead and move forward with synthesizing, now we've incorporated that, and Kosta is going to speak more about the chemistry aspect of making the compounds, and he's going to talk to you about the workflow of making them, and basically you want to test it out. You want to throw it on the boiled egg plot before you even move forward with making the compound, because if it's not in the yolk, then we can rule that out initially. So this was a different layer. This kind of goes to illustrate how complex it is and how difficult it is to find a good candidate molecule to be a good D3 receptor radiotracer. It's a very complex project, and it's great that we figured this out so that we can move forward. As we move forward, we can move forward more efficiently. But it was very interesting to work on that. And we had to initially find out that it was necessary to compete with endogenous dopamine, and then now we've got the so it's
and all of those steps are necessary, but the ordering of them is probably important for the efficiency. So congratulations on the boiled egg, and you can always make egg salad if you have too many. All right. So thank you, Aladdin. Other people's questions before I take up the time here for questions? Sorry, anybody else?
I just had a real quick, and maybe as you mentioned, Aladdin, this will be discussed later on, but so I don't know anything about the boiled egg, but what are just a few of the, and I couldn't tell what the axes there were. So like, what is it about the chemical properties? Like, what do you need to get it into the yolk that you didn't have?
Yeah, so the y-axis, this has to do with the compound's lipophilicity. So they look at a specific type of method for calculating the log P, which determines which is based off of its lipophilicity, W log P. So this is here on the y-axis. So compounds are more or less lipophilic. Then you look at the compound's polarity using specifically this TPSA calculation. Again, Kosta is going to go into this a little bit more in detail because he's, the chemist behind this, and I just kind of understand the axes. But so if a compound, you want it to be you want it to fall its lipophilicity and its polarity within this range. And within this range, because the brain, it's fatty, it has to be a little bit lipophilic. Can't be too lipophilic, can't be not lipophilic enough. And the molecule can't be too polar, and it can't be not polar enough for it to be able to penetrate the brain and get into the brain.
Super. Thank you. Appreciate that.
Absolutely.
Any other questions for Aladdin or comments? I just want to say that this quest is obviously it's not just a years-long quest or even an Aladdin trajectory. It's been going on for some years. The field is really in need of these D3 specific tracers because most of the findings in addiction that you hear about when you hear about brain imaging are using the non-specific tracers. So some of the history of things that have been said about dopamine going up or dopamine receptors down or availability, some of that history will be rewritten once we actually get the specific parsing of these tracers so that we can actually see, for example, that D3 might be going up while D2 is going down, and in fact that it's the ratio or the balance that may be important for medication or for determining vulnerabilities. So there's a lot of exciting things that depend on this. We've all been on the edge of our seats a long time. We've had our fingers crossed a long time, including for Aladdin this week. Okay,
Yeah.
Alain,
Anna Rose, if I could just add a quick point that,
please.
one of the things my group has been doing that I don't think people really spend any time on doing is we spent a lot of time trying to figure out why these compounds have failed. Then, in order to fix them and try to come up with a method that will work. So a lot of what we're seeing is really Everett trying to figure out why these compounds have not worked, and
Right.
that's the first step in figuring out something that will work.
Absolutely. And I like boiled eggs better than I ever did before. Thanks, Bob. And Alain. Okay. Do we want to
Thank
move on
you.
to the next talk? I think we're a couple of minutes into Allie's time, but we'll make it up. So I have the pleasure of introducing Allie Ely, Alice Ely, we call her Allie, who's been actually working with our group in one way or another for almost a decade. She was a Penn undergraduate. She had her advanced degrees at Drexel. She's then at a postdoc with Walter Kay, a very well-known eating disorder researcher at UCSD for a couple of years, and then back to Penn for taking a final year on our T32, working with Reagan Wetherill at the intersection of food and substance use disorders. So, Allie, we are very happy to hear today about your project, which is characterizing the neurobiology of smokers as a function of body mass index. Please go.
Awesome. Thank you so much. I'm excited to share some of this stuff with you guys. I hope you can hear me and see everything okay. Let's see.
Looks fine.
Let's just get organized here. All right. So
on the slide view, yeah.
That's there. So we've talked a little bit about this today already. I'm excited that there's some more sort of transdiagnostic folks coming into the fold. But as many of you know, there's a lot of overlaps between smoking and eating. Current smokers tend to have lower BMIs than non-smokers, but those with longer term and heavier smoking tend to be more likely to have obesity. We also see greater body fat percentage and waist circumference in long-term smokers versus never smokers. There's a lot of really interesting research on the influence of nicotine on appetite and food reward, both in smokers and non-smokers, showing reductions in hedonic hunger, changes in metabolism, both sort of glucose metabolism overall, as well as in specific brain regions. And longer-term smokers tend to have lower reward-related brain responses to food cues when sort of viewing them in the MRI. As it pertains to cessation, there's, of course, both considerable observational research demonstrating post-cessation weight gain, but also we see that treatment-seeking smokers tend to have higher rates of obesity than smokers as a whole or the general population. And there's also greater utilization of smoking cessation medication among obese smokers, or smokers with obesity, I should say. Essentially suggesting that this group this comorbid group may have slightly more difficulty quitting smoking cold turkey on their own or with sort of standard nicotine replacement therapies. And so my focus while I've been in the T32 for the last couple of years has been questioning how BMI influences neurobiology in smokers. And so I'll talk a little bit, my first sort of intro to this was a study that I collaborated with Terry Franklin and Reagan Wetherill in on bold response in smokers, specifically, or ASL bold response in smokers, and we looked at the how BMI influenced brain response to audiovisual smoking cues as compared to non-smoking stimuli, and found that smokers with higher BMIs showed blunted activation in the dorsolateral prefrontal cortex, and that smokers with obesity in particular showed a relationship between this blunted activation and go-no-go response. And this was exciting, and it kind of led to my current position, and now suggested, with all of this observational research, that smoking cessation may be more difficult for smokers with obesity, potentially due to sort of these neurobiological differences and differences in how those brain function differences relate to inhibitory control. So the sort of two big projects I've been working on over the last year or so has been looking at these questions in the context of connectivity and also in terms of self-reported motivation. So I'll talk a little bit about the connectivity study, which is currently in press. It may be official now, but just published in the last few months. And then we can talk a little bit about motivation and then what our next steps are. The question kind of became was, are these differences exclusive to cue response, or is it more of a circuit-wide difference? And so we did this follow-up study looking at resting state connectivity as a function of BMI. We looked at 87 treatment-seeking smokers who underwent fMRI scanning, 31 of whom were lean, 33 of whom had overweight, and 23 of whom had obesity. They were similar across the board for age, education, as well as their rates of smoking, notably. And they didn't differ in terms of their sex or ethnic breakdown. We did an independent components analysis and compared the components we derived with layered resting state network templates to select our components of interest. And so these are the different circuits that we kind of pulled from. The first one that's numbered number 3 for the layered component references, are comprised brain regions that are typically associated with reward network. And I'm kind of speeding through it so I can talk a little bit about specific brain regions if that's of interest. But our salience network, or SN, is the component mark number 4, including bilateral anterior insula and some cingulate regions. Our default mode network component is marked number 13, and along the bottom is sort of the R-derived components as compared to the layered reference components. We also had left and right executive control network components that we also looked at. And what we found was that when we looked at these the functional resting state functional connectivity within these components as it related to BMI, we found that only the salience network had a significant relationship with BMI, where greater BMI was associated with reduced functional connectivity in this network, though this became a trend when we controlled for age and sex. There is some evidence that a relationship between sort of reward response and particularly brain response to cues and some other kinds of rewards is associated with BMI non-linearly. So we also ran a quadratic analysis as it pertained to resting state functional connectivity, and found a quadratic relationship between the default mode network connectivity and body mass index, such that those with sort of with overweight or kind of at the very low end of the quote obese weight range had the highest DMN connectivity amongst all of the participants, where those who were more on the leaner spectrum or had higher BMIs had lower functional connectivity in this circuitry. Lastly, we were interested in how these differences in functional connectivity might be related to actual smoking behavior, and so we ran a multivariate analysis of variance to determine if different groups had different relationships between cigarettes per day, nicotine dependence, and pack years, with the functional connectivity as it was related to different components. And we found that in the salience network in particular, there was this differential relationship between cigarettes per day and connectivity, that differed based on weight status. So those lean participants or those with overweight showed greater, heavier smoking with greater connectivity, whereas the folks with obesity showed the opposite relationship, where the higher their salience network connectivity, the less cigarettes per day they were smoking. We saw a similar breakdown when we looked at nicotine dependence, but not with pack years. And salience network was the only network we saw this differentiation in. So essentially, smokers are showing this reduced connectivity in the salience network with greater BMI. But this differs depending on sort of weight status. And this prior research suggesting that this overweight group might be sort of unique in how it processes rewards or salient, how it processes important information. This group may be sort of more prone to overconsumption. They may demonstrate differences neurobiologically that may sort of support these differences that we see sort of on a behavioral or a self-report level. And I was curious to see how these neurobiological differences, again, sort of translate into sort of self-report, and see if there were differences in smokers' motivations to smoke and how that related to their behavior. So we looked at smoking motivations and how it related to their smoking behavior. The smoking motives questionnaire was given to that same group of treatment-seeking smokers, and that questionnaire is divided into subscales to assess different types of motivations for smoking. Automatic being kind of the conditioned sort of hand-to-mouth without thinking sort of behaviors. Sedative being the relaxing effects of smoking. Addictive being sort of craving focused. Stimulation being sort of the alertness and the effects on attention that come with smoking. Psychosocial motives for smoking being sort of having something to do while talking to somebody, or identity, belongingness, things like that. Indulgent, feeling like you're, sort of that rewarding and feeling of getting a treat or something like that as it relates to having a cigarette. And lastly, the sensory motor aspect of feeling a cigarette between your fingers, or the smell of it, or the feeling of it while smoking. Participants ranged in age from 20 to about 60, and had about 14 years of experience. I'm sorry if there's any background noise here. Groups didn't differ on again, on age, education, or smoking variables. And you can kind of see the breakdown of the ethnic and racial groups in the sample. Notably, weight groups didn't differ in the racial or ethnic breakdown, nor in sex breakdown. And what we found is that BMI was significantly associated with smoking for indulgence, but this was kind of this wasn't a super strong relationship. But particularly, we were interested in how those motivations translated into behavior. And what we found was that, again, there was this sort of dissociation between groups in terms of their relationship between smoking motives and their smoking behavior. Notably, none of these groups differ in terms of their behavior, and none of them differ in terms of their motives, but they differ in terms of how those motives potentially drive their behavior. So for those folks with obesity, there doesn't seem to be a relationship between the motives, their self-reported smoking motives, and how many cigarettes they're smoking per day. But for those with overweight or those in the lean group, we see that the higher sort of they rate addictive smoking motives, the more cigarettes per day they're smoking, which makes sense. This dissociation didn't exist for any of the other smoking motivations, though we did see a relationship within the lean group between, I believe it was sedative and sensory motor motives and their smoking behavior that didn't exist in the other groups. So we see kind of these differential relationships between why people are endorsing smoking and how much they're actually smoking, but smokers with obesity don't seem to be, as influenced by this range of motivations to smoke. And this may be due to the fact that that particular questionnaire doesn't really get into weight concerns. It doesn't get into body image. It doesn't get into trying to avoiding weight gain or managing appetite control. And essentially, my takeaway from this was that we have to be very careful when we're doing these kinds of assessments if we are interested in motivations for smoking or motivations for smoking cessation or things that may get in the way of smoking cessation. We have to take BMI into account in order to fully sort of be able to attend to those motivations and address them in the context of smoking cessation. So essentially, if we're not addressing addictive smoking for folks who are in that lean group, we might not be as effective. Whereas if we're not addressing the motivations to smoke in folks with obesity, then we're not going to see potentially the level of effectiveness that we want to see when it comes to smoking cessation. For our next project, we just started to work on an independent components analysis, looking at how how connectivity changes with menstrual phase and how they might that sort of trajectory may vary based on BMI, as well as looking at the relationship of that to inhibitory control. And then I'm also awaiting feedback on a K award, looking at a model of reward-based decision making in as it relates to BMI in smokers as compared to non-smokers. And I know I kind of raced through to get to finish up before the next person, but I'm happy to answer any questions. I'm excited that there are more people who are kind of looking at this comorbid group and the effects of obesity in smoking and obesity in substance use, and how it might sort of uniquely affect these folks. And I appreciate your attention, and thank you to everybody who's helped out with all of these projects and continues to be of such great support moving forward.
Thanks a lot, Allie. That's a really intriguing intersection that you're working at, and I know that we have crosstalk potentially here with at least 3 investigators within the T32 that will be working at this intersection of substance use disorders and food. And we'll be hearing from one of them later, and we heard from one of them earlier. So we're in very good company here. And, I think I should mention that you also have the K Award under review, right?
Yes, I think it's the 21st that it comes back.
So we're going to have fingers chronically crossed for everyone here.
Yes, for sure.
Congratulations to you on that. And we'll probably hold other questions just so that we don't get too far off kilter here. We're a little bit off, but we'll catch back up.
Great.
All right, so let's move on to our next speaker, be Amelia Dunn, who has worked with Julie Blendy and actually is finishing up the T32. Finished up the T32 on Friday, October 1st, and is heading to a new job, which she can tell you about. You can probe her on that. But today, she's going to talk about her work with Dr. Blendy's lab, talking about acute molecular and long-term behavioral effects of neonatal opioid exposure and withdrawal in mice. Go for it, Amelia.
Okay, thank you. All right. Hopefully those slides are good. Thank you so much for the introduction. It's been really fun listening to everybody's talks today and hearing the updates on everybody's work. As Anna Rose mentioned, I am finishing up my time in the Blendy lab right now. So I'm going to tell you about the study that we have been working on. I've been working on the past 2 years and is ongoing in the lab, looking at some acute and long-term effects of neonatal opioid exposure and withdrawal in a novel mouse model. So I don't think we need to really remind the group that opioid use has really exploded over the past 20, 30 years in the US in all different segments of the population, and that includes people who are pregnant. And so there's also been a corresponding in opioid exposed babies. So this is just a map that was published in 2016 showing the increase in drug exposed babies in local hospitals around the Philadelphia area from 2009 to 2016. And you can see that in 2016.
All right, see you guys. Bye. I can't wait for back with treats. Yeah, let me know if you guys have any other things.
Hey, somebody has a hot mic, so if we could all mute our mics, that would be great.
Thanks. You can see that in some neighborhoods you were getting up to 5, almost 10% of live births had some drug exposure, which is a lot. And so drug exposure during pregnancy can result in neonatal opioid withdrawal syndrome, or NOWS. And so NOWS is a well-characterized condition that includes a whole kind of constellation of symptoms in newborn babies, things like high-pitched crying, lots of gastrointestinal distress, kind of general like fussiness and delayed development, things like that. And so all of these kind of acute effects are pretty well characterized, but much less is known about the long-term effects of NOWS. So you can imagine it's very kind of difficult to study in humans, longitudinal studies of these babies and their outcomes, and then what kind of outcomes are related specifically to NOWS versus other variables in their lives. And so this is where the use of really translationally relevant mouse models is really important. So the Blendy Lab developed a 3-trimester NOWS model. So on the top here, you can think about kind of like the maximum amount of opioid exposure a human baby could experience would be opioid exposure during all 3 trimesters of pregnancy, and then the baby would go through withdrawal at birth. And so the way that the mouse model on the bottom here works is that the mouse is exposed to opioids during gestation, but actually gestation in mice is equivalent only to the first 2 trimesters of human pregnancy in terms of brain development. And so what this model does is it gives opioids additionally after birth for the first 2 weeks of life, so during postnatal days 1 through 14, because that's developmentally equivalent in mice to that last trimester of pregnancy in humans. And so that way we actually measure withdrawal on postnatal day 15 in this animal model. And so those mice are going through withdrawal at a same developmental time point equivalent to humans. So here you can see the way that works is that the mice are exposed to opioids during gestation via a mini pump implanted in the mom. and then through twice daily injections for the first 2 weeks of life. And so I'm going to talk a little bit about some kind of characterization of this model, and then eventually talk about some kind of, like the title of my talk suggests, acute molecular and long-term behavioral effects that we found. So first, we wanted to kind of characterize what this animal model was doing, and compare that to symptoms of NOWs in humans. And so here you're looking at the body weight of the mice during P&D 1, 7, and 14. And so in red, we have the mice that were exposed to morphine throughout development, and in black, the saline controls. And so in both males and females, overall, there's decreased body weight gain throughout these first 2 weeks of life. We also look at the some milestones of development during this period of time. So things like the time it takes for the pups to open their eyes, to be able to walk in a straight line for a certain distance, to be able to flip over onto their stomach and then hold onto a little bar with their forelimbs. And again, same setup here. We have the males and the females, morphine exposed in red and the saline controls in black. And across the board, and we've done this a bunch of different times now, and every time the morphine exposed animals take longer to reach these milestones. So they have delayed development, similar to human babies. Then on postnatal day 15, we measure signs of opioid withdrawal itself. So here we're looking at 8, 24, and 48 hours after the cessation of opioid exposure, so right after that last injection. And here we're looking at the number of ultrasonic vocalizations that the mouse is giving off. So kind of similar or analogous to like the high-pitched crying that a human baby exhibits. And you can see we have a really dramatic increase. This is really consistent at 8, 24, and 48 hours after opioid exposure here. 2 other measurements of withdrawal that we looked at are this hot plate latency, so the time it takes a mouse to exhibit pain on a hot plate. And you can see that these mice have hyperalgesia, so they're more sensitive to pain at both 8 and 24 hours after the last opioid injection. And then we looked at some just kind of somatic signs of withdrawal, so things like climbing on the walls and jumping. And at 24 hours particularly, we see an increase in those somatic signs of withdrawal. So overall, this kind of 3-trimester model of kind of maximum opioid exposure for these pups does demonstrate these kind of decreased body weight, delayed development, and a really robust withdrawal response. So, we have characterized this pretty thoroughly now in the lab, and so we're pretty confident that this is recapitulating something similar to neonatal opioid withdrawal syndrome. And so then we wanted to use this model to ask the question of what are some long-term effects of NOWs. And so we started out by looking at kind of some classic affective and reward behaviors, what we kind of predicted, behaviors that are often affected by withdrawal in adults maybe, or just we associate with opioids in general. And I'm going to blow through these kind of quickly just for the interest of time. But if anybody has any particular questions, we can always go back. I'm also going to blow through them because the data is pretty easy to look at because there are no differences at all. So here we're looking at this elevated zero maze in the mice, and we're looking at time in open arms, which is, a kind of affect on anxiety-like behavior. And you can see in both males and females, and this is in adulthood. So they went through this NOWs paradigm and then we let them grow up. And there's no difference between the saline and the morphine exposed animals. We looked at the tail suspension test that somebody described a little bit earlier. So kind of depressive-like behavior analogous to maybe a forced swim type of test. And again, there's no difference between the morphine exposed and saline exposed in either males or females. Crystal Lemche in the lab did an IV self-administration experiment with these mice in adulthood, looking at remifentanil, so an opioid self-administration. And here you can see the closed icons here are the active lever, and both the saline and the morphine exposed groups learned to self-administer and increased their active nose pokes throughout the session, but there was no difference in the amount of remifentanil administered. And she also did a progressive ratio experiment and again did not find any difference. in the progressive ratio breakpoint as well. And we looked at a couple other affect and reward behaviors as well, and didn't find any differences either. And so then we kind of went back to the beginning because we have this model, and this model is pretty robust. Like I said, we characterized it many times, and we definitely have a very clear withdrawal behavior. And so then we kind of wanted to look at, okay, well, maybe if there aren't that many changes in adulthood, or maybe we're looking at the wrong behaviors in adulthood, let's go back to when we know we have changes in behavior and figure out, see if we can get some clues from what's happening molecularly in the brain. And so we did an RNA sequencing experiment right after gestational exposure. So after the pups were exposed to opioids during gestation, they were born, and then we did bulk sequencing of the brain at PND1. And so you can see here, these are volcano plots. So we're just comparing morphine exposed to saline exposed pups. And you can see immediately that there are many more genes that were differentially expressed in the males than in the females. A lot. And then here you're looking at the fold change on the x-axis and then the significance on the y-axis. And a lot of the genes that were significantly expressed and a lot of the genes that were overlapped between males and females were a lot of kind of metabolism, mitochondria-related genes, and there were also many synapse-related genes as well. So these are the Go terms that were most significantly up and down regulated in females and males here. And so you can see in the upregulated genes, there's a lot of overlap in things like, neurotransmitters, maybe GABA signaling, synapse assembly, things related to kind of synaptic development. Downregulated genes, there's a lot of mitochondrial and metabolism genes as well. So this is a lot of these differentially expressed Go terms pointed to maybe some more classic neurodevelopmental disorders. So things like autism spectrum disorder that maybe aren't as related to things like affect and reward, but might be related to some different kind of long-term changes. So we also compared this set of sequencing results to some known gene sets that are altered in neurodevelopmental disorders like autism spectrum disorder. And so this is just a bunch of different gene sets in the literature that are known to be associated with ASD. And we just looked at enrichment using Fisher's exact tests to see if our differentially expressed genes were enriched for these gene sets. And so you can see that several of these were significantly enriched in our differentially expressed genes, suggesting that maybe there is some sort of overlap in molecular mechanisms between neonatal opioid exposure and other neurodevelopmental disorders. And so then we wanted to look at some behaviors that were related to this. And so the kind of one of the most significant hallmark behaviors of animal models of ASD, at least, is social behaviors. And so we looked at this 3-chamber social assay where you put a mouse in to explore the 3 chambers to acclimate, and then you put in a stimulus mouse, and in our case we actually did a novel object on the other side, and see the change in the amount of time they spend on either side, kind of like almost like a CPP. And so you can see that in our saline controls, we actually didn't see any kind of pro or antisocial behavior. But in the males, our mice that were exposed to opioids actually had a significant social aversion. And so they avoided the chamber with the stimulus mouse. Whereas in the females, we saw the exact opposite. So we actually saw that the females had an increase in social behavior, which was interesting. And then we also wanted to explore some additional behaviors that were related to ASD and other neurodevelopmental disorders. And so one that we looked at was sleep behavior. So if you tuned in earlier for Darryl's talk, he talked about this sleep system where we can track activity and use that as a metric for sleep. And so here we have the amount of time that the mice are sleeping during their dark cycle, which is when they're active, and then the light cycle, which is when they're sleeping. And so you can see that they, have increased sleep during the light cycle, but actually, in the males at least, the morphine exposed mice there was a significant effect of morphine exposure on this kind of general sleep pattern. So they slept a little bit more during their dark cycle. And we had a few other different metrics of sleep that were different in females as well. So it did seem like this neonatal opioid exposure paradigm seems to be altering some long-term behaviors that might be related to other neurodevelopmental disorders like ASD. So kind of moving forward, what we want to do is investigate what are there some molecular mechanisms that are similar between NOWs and other neurodevelopmental disorders. So from our sequencing results, we know things like synaptic development, nuclear export popped up a lot, metabolism, that those things have all been implicated in neurodevelopmental disorders and now also in NOWs. Things like immune system dysfunction, potentially changes in the microbiome are all molecular mechanisms that would be interesting to explore. And then finally, we're also interested in looking at whether there's additional overlap in the behavioral effects of NOWs and other neurodevelopmental disorders. So investigating further changes in social behavior, things like repetitive behaviors, like repetitive grooming, for example, measures of cognition, and also we're especially interested, all of the behaviors I showed were during adulthood. And so kind of an immediate next step in the lab is to investigate some of these changes in adolescence as well. So yeah, it was very exciting to work on this project. I had a really wonderful time in the Blendy lab, and I'm sad to be leaving. And I'm, as Anna Rose mentioned, moving on, looking for jobs in kind of healthcare, pharma communications, and industry now. And so I just want to thank the T32. It was a really great experience to be able to learn so much clinical science as well. That was really helpful. And I'm looking forward to hearing the rest of the talks this afternoon too. So please let me know if you have questions. Thank you.
Thanks a bunch. And we would love to have any questions that people have. We're just a few seconds to the good, so we made up our time. Thanks for that, Amelia.
Talk fast, yeah.
No, it's very coherent, very smooth. And, you know, when we have more time, I'll ask about these interesting sex differences where things go in opposite directions. Just very, very interesting. But
yeah, and the fact that actually both in the social behaviors and in the sleep data, the females seem to have kind of an adaptive response, which is really interesting and something that we're excited for further.
It's quite interesting, and I think, you know, NIH keeps saying they want you to be able to analyze your results by sex, and so sometimes they're not willing to give you the budget to do that. But they do have the right question, is that these things are sometimes very different, and if we don't look, we can't find it out.
Yeah, definitely. And there's lots of human sex differences too, so that's another really interesting overlap.
Absolutely. Any other questions today for Amelia? Amelia is my daughter's name too, so I say it with great affection.
It's a good name. Feel free to message me there were a couple questions in the chat, but I can maybe just respond to the chat if that's
That's okay, Amelia, just type it in.
Okay.
It's Liz. Sorry. I'm here. Yeah, just type it in or we can talk offline.
Okay, yeah, sounds good. Thank you.
That's great. Okay, so I think we'll move to our next speaker. And our next speaker is Konstantinos, otherwise known as Costa Plakis, who actually completed his pre-Bob Mock work at SUNY Buffalo. And I think that's where Bob Mock completed his graduate work as well. But that's not why he chose him for the lab. He chose him because he could recognize that he was a chemist with hot potential. And in fact, what he's been doing with Bob is actually working around very specific mu-opioid receptor agents, possibly antagonists, but PET tracers that will really help us advance the field. As many of you know, we basically have a lot of opioid work, but very few tracers that don't involve carfentanil. There are a handful, but not too many. And the highly potent carfentanil that you hear about in all the public press these days as being something that can cause overdose, well, it's also a pain in the neck to work with as a brain tracer, because you have to be very careful, because just a very little bit of it can in fact have pharmacologic effects. So it would be so wonderful to have some highly specific opioid receptor agents that act as tracers, and who knows, maybe as therapeutics, to reduce the overdose effects of very potent agonists like fentanyl in the future. So Costa is going to tell us about this. Please go ahead, Costa.
Thank you very much for the kind introduction. I just wanted to double check that everyone can see my slides and can hear me.
I can. Yep. I think they're there.
Excellent, excellent. Just a quick disclaimer, my power just went out, so I'm using a hotspot through my mobile phone. So
jeez.
if I suddenly drop out, perhaps you can send some smoke signals to Northeast Philly.
Is this a weather related event or just a Costa related event?
What's this? Murphy's Law? What can go wrong will go wrong. I think it's the latter. Maybe this is because I started on April 1st, you know.
Could be.
I think I made that joke once before. But I'll get right into it. Thank you for the introduction. And so moving right along, my slides are a little slow. Many of you are probably familiar with these statistics, and these are the most recent I've been able to find from the CDC, which cite approximately 50,000 deaths involved opioids during the calendar year 2019. Some preliminary data that I've seen suggests that 2020 is going to be even worse. As you can imagine, we've had a lot of crazy things going on during calendar year 2020. So I've seen that number could be as high as 90,000, which is definitely the wrong way we'd like this curve to go. And I think one of the key takeaways here is that we urgently need more tools to both treat and image patients suffering from opioid use disorder. And from a chemist's perspective, a medicinal chemist perspective, one of the problems is that medicine can generally take about 10 years to translate from bench to bedside. And so we could do the math if we wanted to, 50,000 deaths annually for 2019, that's 135 or so per day, times 365 days a year, times 10 years, that's an astronomical cost of human life that I think we need to start rethinking how we go about designing drugs. And one way that I've kind of recently come to realize might be useful is through drug repurposing. And so through scouring some of the literature with the direction and help of Tom Graham. He's kind of been my godfather. He's got a little bit of an eye for this sort of stuff. But we've been able to identify this molecule developed by GSK over the last 10 years or so. And so this is GSK 152-1498. It has underwent several clinical trials, 5 phase 1 and 1 phase 2. It's been used to treat those suffering from eating disorders and alcohol use disorders, and it shows a pretty good safety profile, well tolerated up to about 100 milligrams dosage. And more interestingly, it's both potent and selective for the mu-opioid receptor. And so this is some data presented from GSK GlaxoSmithKline in which they compare naltrexone, its key metabolite 6-beta naltrexol, their affinities for the mu, kappa, and delta opioid receptors, compare and contrast with GSK 152-1498. So you can see here we've got about 1.5 nanomolar as our binding affinity for the mu opioid receptor, and it's suggestive that we have high selectivity over both the kappa and the delta opioid receptor. So this got me thinking with, a favorable safety profile and the fact that it is both potent and selective and mu-opioid receptor antagonist. Could this be perhaps repurposed to treat those suffering from opioid use disorder? I think given the fact that it's already underwent several clinical trials, its translation might be quite rapid. So maybe we can cut down that 10-year timeline to something fewer than 10 years. And I'm also kind of on the lookout for other molecules like this in the literature. And I've identified a couple that I won't get to present on, but happy to chat offline if anyone is curious. Some of the key findings that the teams from GSK have put forward in the literature, and it's not a lot of data because they're kind of hesitant to put all the good stuff out there, as I'll talk about momentarily, but they were able to demonstrate that GSK 152-1498 is indeed an inverse agonist that will act on a constitutively active mu-opioid receptor and decrease its percent maximal response, especially in comparison to known ligands like DAMGO, and that's both in human and mouse mu-opioid receptor expressing cells. And it affects behavior. So this is in a mouse now. Treatment with GSK 152-1498 leads to a dose-dependent caloric deficit, which in turn leads to weight reduction, which is pretty interesting. So that perhaps suggests that it does indeed alter behavior. So with that in mind, I'll just list a couple of project goals that I've been able to think that would be worthwhile that we pursue. One, as I've alluded to, is perhaps we can investigate the repurposing of this molecule from treating those suffering with alcohol use disorder and eating disorder to treating those that suffer from opioid use disorder. Again, its favorable safety properties may enable its rapid clinical translation such that we can get a tool to the clinic quickly to help those suffering from this disease. The second goal, and this is what I'll spend a great deal of time speaking about today, is I'm interested in exploring chemical space around this molecule through an SAR campaign, and that's chemist shorthand for Structure Activity Relationship Campaign. With the main goal, or sub-goal, being that I'm looking for a radio tracer to replace C11 carfentanil as kind of the gold standard for mu-opioid receptor imaging. So we have some structures on the left. Here's carfentanil on the bottom, which again is currently used clinically to image the mu-opioid receptor. And we have a different molecule up top, and this is the one I'm interested in, the GSK molecule. The teams at GSK have demonstrated that this molecule can get into brain, so this is a blocking study where they've administered C11 carfentanil and demonstrated that you can block it with an oral 50 milligram dose of GSK 152-1498. And this is something that is an issue replacing C11 carfentanil because of what Anna Rose alluded to previously. It's somewhat of a dangerous molecule that carries with it certain liabilities when we use it. So it can lead to addiction, respiratory depression, and often requires a lot of licensing to even be able to use. Whereas something that is an antagonist might be a little bit more user-friendly and certainly more safe to use. The first thing I did when I got in the lab and kind of got interested in this molecule is I wanted to really get an idea of what other molecules related to this molecule on the left had been made, had been characterized well in the literature. So I made use of this database called Chembl. And Chembl is great because you can peruse drug targets, in this case the mu-opioid receptor, and you can pull out files that list molecules that are known to interact with it. And so I did that, and then Tom Graham in our lab was able to write some code that allows us to score by similarity molecules known in the literature to the molecule that I was interested in interrogating. And so what we could see is that we have a similarity score reported here, which ranges between 0 and 1. The closer the similarity score is to 1, the closer structurally that molecule is to the GSK molecule, and the closer it is to 0, the more dissimilar the two molecules are. You can see our closest hit here is only 0.23, which to me suggests that there's a vast amount of chemical space to explore, and perhaps that also means there's a vast amount of antagonists that we can make that are both specific and selective for the mu-opioid receptor. So a lot of space to move into. So I'll spare everybody the nitty-gritty details of a synthetic scheme, but suffice it to say there are a couple of key hallmarks that I just want to make you all aware of for an SAR or a structure-activity relationship synthetic campaign. One is that the goal is to be able to generate lots of molecules to get into the assay from a relatively modest number of precursors. And so this is starting from a commercially available bromobenzamide precursor. And over the course of 1, 2, 3, 4, 5 or so steps, we're able to get that one molecule into nearly 8 molecules that we can screen in-house. This was developed by the team at GSK for a large-scale synthesis. So if we ever need a lot of this, that's great. I mean, it works pretty robustly. There have been some challenges associated with its scale down, namely this reaction here. So going from 9A to compounds 10A all the way through 11D. This is a reductive amination, which when developed for a large scale can make use of a very obscure piece of glassware called the Dean-Stark apparatus. Won't go into the details, but suffice it to say, I had to get creative in finding a way to sequester water in order to get this yield up to a reasonable amount. And so I've been able to do that through the addition of molecular sieves. And so what we're doing here is making lots of different molecules that get into the assay, where what we're changing is this R1 and R2 group. So R1 can either be, in this instance, the fluoro group or hydrogen, and R2 can be one of these various substituted amines. So with that, we've been able to synthesize a total of 16 compounds that I've been able to get into our assay with the help of Aladdin Riyad, who you heard from earlier, who's been instrumental in developing this diphrenorphine competition assay. And these Ki values you see are an N of 2. And Aladdin's been kind enough to even let me kind of, tag along in lab and see how these things are done and analyze the data. So I'll give a special shout out to him for his help. But what we can see, before we get to what we could see, I'll also mention we're probing these molecules for their intrinsic activity. So we're submitting them to the psychoactive drug screening program in the Roth lab at UNC. And we have some interesting hits. So we have a range of molecules that bind with an affinity of about 3.2 to 11.2 nanomolar. And so these are some candidates that we're interested in advancing to see if we can perhaps install a radiolabel and then use them for mu-opioid receptor imaging. One way we can streamline our synthesis and pick molecules to synthesize that we think are going to be having the highest chance of success is through the use of this boiled egg plot that Aladdin had presented on previously. So it's got a very friendly graphical user interface, and it's honestly kind of cute and nice to use. But essentially what this is going to track is the W log P, which is a measure of the octanol water coefficient on the y-axis. And you can think about this as determining a molecule's probability of getting stuck in some sort of a lipid bilayer. And on the x-axis, we'll have its topological polar surface area. So that's an idea of how sticky this might be, how many hydrogen bonds it has, etc. And so it's pretty promising, right? We have a lot of molecules in the yolk, and just as a friendly reminder, the yolk, or the yellow, is predicted to have blood-brain barrier passive permeability. So most of these are indeed BBB penetrant, which is good, especially from a PET tracer development point of view. It means that they should be able to image in the brain the mu-opioid receptor. I also think about it as a little bit of a double-edged sword, though, because it doesn't really help me limit my synthesis, but I suppose 14 leads is better than 0. So we can see we have quite a bit of room to play with here. Many of these compounds are in the yolk, so we have some functional group tolerability, which leads me to my next part of the talk, where we talk about installing a functional group to enable the radiolabeling of this compound. So there are a couple of rules we want to keep in mind. Traditionally, this is done by taking the lead molecule, installing some sort of functional group, which then enables the radiolabel to be put on it, ideally in the last step or penultimate step, so we're not handling a lot of hot material. Also, so that your tracer is of high enough radioactivity when it's injected. Ideally, this is done in a manner to allow both the screening of the cold and the hot compound. And if we can keep the structure similar, if not the same, to GSK 152-1498, that would be great as well. A couple of other rules we'll just keep in the back of our mind, right? So classic Lipinski's rules still apply: molecular weight less than 500 grams per mole, perhaps fewer than 5 hydrogen bond donors, less than 10 acceptors, etc. So I designed this scaffold to be compatible with some novel radiolabeling methodology that Tom from our group had a chance to kind of work out whilst he was kind of transitioning from Merck back here, and also done in conjunction with the Macmillan group back over in Princeton. So what we're going to do here is we're going to start with a similar starting material, the substituted bromobenzimide, but we're just going to replace the hydrogen that was here with either a methyl or a bromo. We're going to carry that starting material through our synthetic pathway. And what we're going to get at the end are compounds 5A through 6D, where the class 5 has an R1 equal to the methyl, and class 6 has an R1 equal to the bromo. What this enables us to do is to take the class 5 group of molecules where the R1 is the methyl. What we'll do is we'll screen them in-house, identify molecules that we think are interesting, characterize their binding, perhaps send them out to be screened for intrinsic activity. And so, for example, let's say we found 5D as having some interesting properties. What we'll do then is we'll use this novel methodology, and we'll take 6D And now we have the option. We can either do carbon-11 or tritium labeling as a function of this new interesting methodology. So this is organometallo-photoredox chemistry, which is, I think, very fascinating. So it's using metal catalysts and light to generate new carbon-carbon bonds. And this allows us to install a hot atom, either carbon-11 or tritium in the last step as putative tracers to be used in mu-opioid receptor imaging. So this is sort of where the project stands right now. I haven't finished making compounds 5A through 5D yet, but that's probably good because we can use our boiled egg plot to predict and prioritize our time as to what might be the most interesting and worthwhile to synthesize. So compound 5D, you can see, is kind of just on the cusp of being blood-brain barrier penetrant. So if we were constrained for resources, we can perhaps limit our synthesis just to 5D, although having isolated all immediate precursors, I should be able to just isolate 5A, 5B, 5C, and 5D, screen in-house, and see what their binding affinity is. A little bit surprising that the installation of this methyl group does seem to shift the log P northwards or upwards quite a bit outside of the yolk. But thinking recursively, we can use some of the data from my initial 16 compounds to think forward and make new compounds that are inside the yolk now. So these are still to be made, to be synthesized molecules, compounds 13A through D, and what we've done is just remove the methyl group, and so maybe we can kind of illustrate this. Going from 5A, for example, to 13A, all we've done is remove the fluoro group, and that seems to drop the log P sufficiently enough that is now inside the yolk, and this should be blood-brain barrier penetrant. So I'll wrap up with a quick summary. Through scouring the literature, we found an interesting molecule that we think could perhaps be repurposed to treat those suffering from opioid use disorder. The structure-activity relationship campaign is ongoing to determine molecules or to find molecules that bind with high affinity and high specificity to the mu-opioid receptor that we can then elaborate into a tracer. And so as of right now, I'm most interested, most excited about compounds 1D and 13D. Their screening in-house to determine affinity and perhaps elaboration into either carbon-11 or tritium analogs to discover their potential use as a tracer. So I saved the most important slide for last. I'll just give a quick shout out and acknowledge the support of my advisor, Bob Mack, who's been great, supportive, provided me a lot of resources to get the work done here, as well as some beneficial instruction. Tom Graham and CJ and the group have been awesome in helping to get some of the in silico work up and running. So I didn't get to talk about, we're also interested in streamlining this through the use of free energy perturbation theory. Aladdin Riyad, who you heard from earlier, has been kind enough to allow me to follow him around the binding lab to learn more about the in vivo and in vitro assays, so I can talk a bit more confidently about the data. The folks over at the P30 and T32, Anna Rose and Julie, the funding source, the NIH, National Institute of Drug Abuse. And with that, I'd be happy to answer any questions. And thank you all for your time.
That was really fun tour de force, and I really appreciate it, Costa. And you know, when I think that, are you sure you've only been here since April? You guys have really had a lot of fun in a fairly short time, because I remember when Thomas wrote the email and saying, you know, whatever happened to GSK 1521498? And we found that NIDA had just sort of lost enthusiasm, and there's a long story behind it, but basically nothing was wrong with it, is what it comes down to. It was safe in humans. It had this really interesting profile. And look at this. You've used this as a springboard and have found some things that look really cool. And what with the in silico techniques with Kemble and the boiled egg, it seems like the gates are really open for you guys.
Absolutely. It definitely helps streamline the work, and it's also a great opportunity to learn. So I'm a classically trained organic chemist, but being able to come here and, you know, kind of work with computers to help streamline the process, it's been a blast. A bit intimidating at times, I won't lie, but Tom and CJ are patient and kind enough to allow me to, you know, make a couple mistakes. Then I could ask them for help, and they point me in the right direction.
It's a great team. Does anyone else have any probing chemistry questions? Do all those formulas look all right?
No carbons, but too many bonds.
If not, we will thank you very much, Costa, and I'll move on to the next presenter. All right. And for our next presenter, we're still in the opioid world. We have Greg Salamando, who's been here a little shorter time than Costa. He actually, in the T32, but he was with Greg Corder's lab, coming there from Vanderbilt over a year ago, and we invited him onto the T32, and yet, when did he start? Yes, that would be Friday. So, last Friday. So, he probably will be reporting a progress that occurred since before Friday, but nonetheless, it'll be impressive, and it's in the opioid domain. Take it away, Greg.
Thank you, Dr. Childress. Can everyone hear me all right? Is everything good? You can see my screen? Okay, I'll take that as a yes. Yep. So
Yes.
thank you again for the introduction, Dr. Childress. It's nice seeing everybody too, and getting to, even though we're not together and missing out on the supposedly wonderful chocolate torte, which I'm very disappointed about, but hopefully for next year. But yes, as she mentioned, I just got appointed, but I've been in Dr. Kaur's lab for a little while. And I'm just going to tell everyone today a little bit about what we've been doing in Greg's lab to start working with this new molecular toolkit of viruses that we've been designing to interrogate opioidergic circuitry and pain and look at pain-related disorders. So there's a picture of me again, nice little headshot from Vanderbilt. But yeah, just as Dr. Childress mentioned, just give a little background on myself. I completed my PhD work with Dr. Danny Winder back at Vanderbilt, studying the bed nucleus of the stria terminalis and MDA receptor-regulated excitatory signaling in the region. Now I've obviously moved on to Penn, working with someone who's also named Greg, but this time not on the BNST and on affective disorders again, but now looking at pain disorders and the intersection of that with affective disorders and motivated behavior, as well as looking into, as I mentioned, opioidergic circuitry and how we can utilize different kinds of tools to answer important questions about this circuitry, especially in regards to different behavioral and pathologies related to opioid abuse. And so just quickly, I'll just mention, Dr. Childress mentioned I could maybe just quickly give everyone a little flavor of my background and my training. But this is more or less the paper that came out from my work with Dr. Winder. If anyone's interested, you can take a look at it on JNeuro's website. But essentially, I'm more classically trained in ex vivo electrophysiology, transgenic mouse modeling, and behavioral modeling using primarily mouse models. And I've also done a little bit of work with in vivo fibrophotometry, and I'll kind of pepper into a little bit of what I'm going to talk about in terms of where I'm going with my work in Greg's lab now. So again, obviously, we've belabored this point a little bit in the rest of the talks, but I figured I'd have them there. So obviously, everyone in this group knows that the opioid addiction problem is a huge issue both in the US and globally, with as some of these more recent figures have shown, upwards of 90 plus deaths a day. And as Costa and other people had correctly mentioned, tens of thousands of deaths in the past couple years or so. So it's certainly a problem that's in need of new and interesting treatment methodologies to try and circumvent the classical opioid-based therapeutics that we're using right now. And so in Dr. Kaur's lab, that's one of the questions that we're trying to address from a more biological angle, which is, are there ways that we can identify and then manipulate emerging opioidergic cell populations and circuits, both within and outside classical reward networks when we're talking about opioid-based addiction, and as well as circuits that might be involved in driving nociceptive-like behavior, with the ultimate goal of developing and testing novel tools now for probing these things preclinically and then addressing whether or not these might be able to translate over to the clinic to actually help patients on that side of the equation, which is ultimately the goal of a lot of our work in this group, I would say. And so one of the ways that we're trying to get at this question now from a different angle is, as my talk title suggested, developing a toolkit of adeno-associated or AAV viruses that can directly target either some of these pain-responsive or mu opioid receptor specifically positive expressing cells. And we've done this by working together with collaborators over in Stanford to develop these viral constructs that utilize the mu opioid receptor promoter to drive the expression of a number of different activators, epifluorescent tags, and other kinds of molecular tools that allow us to measure activity within neurons. So in this case, we've developed suites of tools that have DREAD receptors that can be encoded and driven by the mu-opioid receptor promoter, opsins, fluorescent tags, and recently we've had some help developing a GECI, or a genetically encoded calcium indicator, that can allow us to look at putative activity of these cells in awake and behaving mice. So again, this work is kind of all the brainchild and has lent enormously to the efforts of Dr. Charu Ramakrishnan out at Carl Deiseroff's group in Stanford. And essentially what she's done is she's developed a couple of these basic backbone constructs that are running off of either the murine or the mouse mu-opioid receptor promoter, or the human based mu-opioid receptor promoter. Again, these are the two most, I guess you could say, vanilla flavors of the constructs that we have and we're initially starting to work with simply just encode an EYFP tag. And Dr. Ramakrishnan did some initial screening of these viruses in her lab using rat primary hippocampal cell cultures to basically show proof of concept that they are able to be to transfect neurons in vitro and get into cells. And now, kind of after her work, and she shunted some of this over to us, it's come onto our lab to now do some further implementation with these tools to see if we can actually get them to work in mammalian species of interest for preclinical research into opioid behavior and pain behavior. So as Costa and some other folks have mentioned before, in terms of finding putative targets and areas of interest, of course, there's a wealth of literature, both in the preclinical animal modeling field as well as in the human literature, of regions of the brain of interest that are of interest in regards to their responsiveness to both the binding and the activity of opioids, as well as to regulating specific behaviors and maladaptive behaviors that are associated with either pain or opioid-based abuse. So here you can see some representative binding assay images overlaid on MRI, functional MRI images of patients that were scanned while undergoing emotional and perceptive-based tasks. And what you can see is some overlaps in a couple key areas of interest. Some folks have mentioned this a little, but specifically into our lab, we've been focusing on regions like the anterior cingulate portion of the cortex, and as well as the amygdala, which you can see in the far right image there, with the idea of obviously trying to translate this human work into what we've seen preclinically and ask where are some targets that we can initially start utilizing to test these viruses and see if they have any traction and potential usefulness in studying these selected populations and circuits. So here's just kind of a quick schematic of what I've been doing for the past, I guess, almost about a year or so in Greg's lab, both on my own and in tandem with a number of excellent collaborators that we're fortunate enough to have here at Penn. So on the mouse side of things, since our lab is primarily a mouse research lab and does a lot of behavioral modeling and work in these animals, I've been working to both validate the expression and the functional viability of a number of these viruses by injecting them into several regions that we know to have putative mu-opioid receptor positive cell populations, including the anterior cingulate cortex and the central amygdala, as I mentioned earlier, as well as some other areas such as the ventral tegmental region and the periaqueductal gray. And in concert with this, we've also been trying to do some complementarity studies to see if this expression extends across other mammalian species of interest in preclinical research, primarily shrew and rats, which we've been working with Dr. Tito Borner from the Hage and Schmidt labs. He's been helping doing a lot of the surgeries on that and assisting with the histology to see if we can see expression of these viruses in these species. And then with Herculean effort and help from Dr. Sebastian Tremblay from Michael Platt's group and his macaque research interest group, we've been doing similar injections and testing in macaque monkeys as well with their assistance. So next couple of slides, I'll just show you a couple of the images. This is just some basic IHC, looking at how these viruses are expressing across some of these regions in a few of these sample species that we've been looking into. These first images are from mice. And as you can see, with some initial injections I've done in the ACC, as well as the CEA and the VTA, we can see pretty robust expression of this virus, actually. This is using the mouse mu-opioid promoter-based virus in this image right here. And as you can see, at some higher magnifications, when we're looking at where the virus is expressing across these areas, we see nice overlap of our EYFP signal that is indicating our transfected cells that the virus got into, overlays very nicely with a number of neurons that are marked with the NUN neural cell body marker here. And that extends across a number of these areas. So it does seem like these viruses have viability at the basic level with just the epifluorescent tag, at least, in mouse species, which is, of course, great for me because that means I can hopefully move forward using a lot of them for some of the more complicated things we're going to get into in the next couple months. From Dr. Boner's contribution, just showing some of the rat injections that he's conducted so far. We have seen similar levels of good expression and good transfection rates of cells within and around the CEA with the same viruses. And again, once more, you can see in these magnified images, we have some nice overlay of our signal with the neural markers of interest. So it does seem like these the virus is getting more specifically into neurons, not into surrounding glial cells and other non-neuronal cells within these regions. And lastly, here are some images from our macaque injections. We were fortunate enough to be able to work with a single monkey last year that Dr. Tremblay helped us to inject into several different regions. I'm just showing a couple of some of the more, I guess, lovely transfection efficiency areas that we were able to get from this work. On the left here, you can see transfection of the virus. This time it's the human promoter-based variant of the virus into the ACC of the macaque monkey. And we had some very nice stratification of cell types that we were getting, or cell bodies, I should say, that we were getting labeled with our virus that seemed to translate well over to where we believe some of these putative mu-opioid receptor positive cells are in terms of their layers with the cortex. And the signal, once again, overlays very nicely with our new N marker, indicating that we are getting into these cell types of interest here. And on the right, we have another set of images, this time in the CEA, or in the amygdaloid structure of the macaque monkey, this time with the mouse variant once again, showing similar levels of high transfection efficiency in this area. So that was certainly a breath of fresh air for us because it was a long time going with those macaque studies, and it would have been kind of a bummer if it didn't work out. So big thanks to Dr. Tremblay again for his help on that, and Michael Platt's group. So moving from there, what we're trying to do next is, of course, it's nice to generate pretty pictures and see that we have good transfection of our virus at the level of protein within these cells. But of course, we want to make sure that we are actually getting good expression of our virus in the mu-opioid receptor positive population that we're really interested in targeting with them. And so one of the ways that we've been trying to assess this is by the use of in situ hybridization. In this case, a set of reagents and a technique known as RNA scope in order to look and see if transcripts for the EYFP signal that should have been transfected along with the virus into these cells overlays well with the native signal for the OPRM1 transcript, which encodes the mu-opioid receptor. So you can see in some of the images here, when we're looking at our transfection levels and the staining of these transcripts of interest in and around the CEA here, just using this region as an example of this. When we look closer and parse out some of the overlaying cell populations, we can see pretty decent overlap of the EYFP transcript signal with the OPRM1 transcript signal. It's obviously not 100%. Could be a little bit of leak of the virus, as well as a number of OPRM1 cells that aren't going to be transfected by the virus based on both how accurate our aim is and both at the efficiency level endogenously of the virus. But on the whole, when counting just the EYFP positive and the OPRM1 pre-positive cell overall, this was done over the course of 2 to 3 mice, counting both the left and right CEA in these animals. We can see that we have a pretty decent level of overlap between our signals that we're probing for. Around like 75 to 80 to 90% or so of these cells seem to have a good correlation with both of our signals of interest. So hopefully that's indicative that this virus is indeed working the way that we would hope them to, and it is getting into the cells of interest that we want to then probe and look at further down the road. In addition to this, we're also trying to, of course, functionally validate the effectiveness of these viruses by also looking at the physiological properties of these neurons and trying to probe some basic changes in synaptic and cellular physiology that we would expect to see if these were indeed cells that express the mu-opioid receptor. And so here I'm just showing a couple examples of what we've been kind of spinning up using patch clamp whole cell electrophysiology in this group, in our group to try and validate this. Here, obviously, the goal is to patch onto these mu-opioid receptor cells that have been tagged with our EFYP virus, and then to basically what I've done is try and assess whether or not they're responsive to mu-opioid receptor agonists and other compounds that are selective for this receptor. In this case, some of these basic first experiments I've been trying to do has been just to wash on the agonist DAMGO and see if we have a stereotypical response that we would expect in mu-opioid positive cells to binding of that drug onto the receptors, in this case, which would be a reduction of AP firing and an increase in overall cell membrane potential, making it harder for the cell to fire due to the nature of how the mu-opioid receptor is acting at a biochemical level in these cells. And so here's just a couple quick examples of what I've been seeing so far. This work is ongoing. I actually have a batch of these mice ready to go for this week, starting tomorrow to start doing some more of these tests and flesh out the physiology a bit more. But a couple initial first passes, we were able to see that indeed when we patched onto these EYFP positive cells and washed on our DAMGO agonist, we did indeed see an increase in the rheobase, which is the rough membrane potential around which the cell first initiate a action potential firing. A higher rheobase usually indicates that the cell membrane potential has increased, making it harder to get to it, or sorry, decrease and making it harder for it to get to a firing potential. And then as we see over here, when we're looking at the AP firing frequency, we did see a precipitous drop in the overall number of action potentials that occurred following DAMGO hitting this bath and then getting onto the cells. And that this effect did not wash out when we reapplied just generic ECSF onto the slice. So that work, as I mentioned, is further ongoing, but there's a couple other things that we're going to be trying to do too. Aside from just validating these viruses, of course, the end goal of this is to see if we can answer relevant biological questions regarding opioidergic and pain-responsive cell population and cell circuitry. And so one of the ways that we've been starting to look into this is through another project that I've been starting to put together in the lab, which has been looking at a circuit that was discovered projecting from the anterior insular cortex to the basolateral amygdala, initially around the early 2000s from the lab of Dr. Peter O'Hara. As you can see here are some inserts from their 2003 paper in which they first started to probe and look into this circuit in rats. And essentially what the group was able to see was that if this putative population of glutamatergic cells that they discovered in the rostral and agranular insular cortex was inhibited, it seemed to increase analgesic-like behavior in the rats, leading them to believe that if these cells were disinhibited or there was any change in tonic inhibitory activity in the cells, so this could potentially lead to the opposite kind of phenotype in the animals. So this has led us to believe and led us to be interested to follow up on their work now using some of the mouse models that we have available to us to see just how this input might be shaping pain and pain perception in rodents, and ask other questions that are pertinent to the cell and circuit specific effects in terms of how they're regulating effective and physiological responses to pain. And so just to quickly go over what I've been doing so far, again, since this work was first done in rats, I wanted to follow up on this and see if I could validate that the circuit did indeed exist in mice as well. So using a viral strategy here to inject a retrograde Cree reporter virus into the BLA, and then a Cree-dependent reporter virus into the AIC, what I was able to see is that indeed there does seem to be a projection that exists from these two regions, which allowed the M-cherry fluorophore hue to be expressed within the anterior insula of the mouse cortex. And then on the right over here, another kind of validation study we did was to see how many of the cells in these areas in this region of the AIC might indeed be opioidergic or express the mu-opioid receptor in this case on them. And using another viral strategy here, in this case, a new virus that utilizes a nuclear localized M-cherry signal as well as an EGFP signal that is responsive to the presence of Cree in a cell. What we did was inject an OPRM1 Cree transgenic animal. I believe some other folks in the previous talks had mentioned the use of this animal model. But essentially, mice that have the Cree expressed in them will be driven by the expression of the mu-opioid receptor promoter. So only cells that have Cree in them, when hit with this virus, will show the EGFP signal, whereas other cells that are transfected and do not express the Cree recombinase will just express the M-cherry signal. So as you can see here, here's validation of our viral injection in and around the BLA region. And when we looked up in the AIC, we were indeed able to find a number of pre-positive neurons, as well as a few M-cherry positive cells within this area, which may seem to indicate that there is indeed projections of opioidergic-based cells from the AIC down into the BLA. And so this, of course, kind of opens the door to what we might do next, which in this case would be me attacking these two projects from a two-pronged approach. At the moment, I'm still in the weeds with finalizing some of the testing and validation of our viral toolkit here, as I mentioned before, continuing on with some of my physiological studies to again show that when doing slice work, we can indeed get the kind of period of responses we would expect to see from these more positive cells when assessing their synaptic physiology. And we're also moving into starting to work with our GECI encoding the opioid receptor promoter virus, doing some in vivo photometry work to see if we can get responsive activation from these cells that are transfected with the virus in response to basic nocturnal stimuli, as well as potentially other responses and behaviors more relevant to motivated behavior and opioid self-administration or dosing. And then down here, we're starting to move on to further investigate this initial sample circuit and see if we can get some basic responses and changes in the behavioral profile of the mice that replicate what the original study that I mentioned back in 2003 showed, and then go in using our more specific viruses to see if we can replicate those effects by specifically manipulating and altering the activation or measuring the activation of the mu-opioid receptor specific cells that we have found and identified in the AIC here. And lastly, of course, I'll just make a mention that the goal of this work is obviously just to start to investigate the possibility that the use of these constructs may be feasible as either a potential gene therapy-based approach or to basically broach the topic of using something alternatively to standard pharmacological approaches to manipulate these cell populations and see if there's an avenue that can be open to look into alternate means of treatment for both studying and regulating these kind of circuits in a patient population. And so with that, I'll close out by thanking everyone for their time. It's really great to see all the work that's going on in this group and to be part of this group of scholars. I'm really excited to see what happens with the continuing work of all the trainees here in the next couple of years. And I'd like to, of course, extend thank you to all the folks in my lab for helping get this project up and running. Our collaborators are Michael Platt, Pete Schmidt and Matt Hayes' group, as well as Bart Dijong's group, Carl Deitzeroth's group out in Stanford, as well as all the wonderful members of the T32 Training Advisory here, and of course all the support that Greg and myself now will be party to. So thank you guys for your time, and I'm happy to answer any questions.
So much, Greg. Because we're running a little bit past time, I will.
Sure, sorry about that.
That's okay. I'll have to ask people to hold their questions, but you may indeed have some, so watch your chat. And also give a shout out to Greg Corder, who will actually be giving a talk to our group on November 1st. And so we look forward to that, and we'll hear more about your work and his on the 1st of November. All right, let's go to our next speaker. And we have Dr. Cindy Lee, who is going to be speaking to us about the role of obesity in altering the neurobiology of neurobiological responses to morphine withdrawal. A little bit about Cindy very quickly. She comes to us from Rutgers. She's going to be studying with Corinne Weirs, and basically, who knows what's the future holding, but right now we're going to hear about her work that she's just been doing, because she too, yes, joined the T32 3 days ago, just like Greg. So this will be her work leading up to her T32 projects, and you'll hear about those next year. Dear Cindy, please let us hear. You might want to try the side view. Perfect. Thank you.
You're on mute, please. Cindy, you're on mute.
Can you hear me now?
Yes, all is good.
Yeah, I just want to say thank you for the opportunity to be a part of Penn, to be a T32 fellow. I'm really excited for this opportunity, but in the meantime, I'm going to be sharing some of my dissertation work. focusing on the role of obesity in altering neurobiological responses to morphine withdrawal. So obesity and opioid abuse are two major public health problems, and there are some evidence suggesting an interrelationship between the two disorders. One study showed a positive correlation between BMI and prevalence of long-term prescription opioid use. And we attributed this to the increased prevalence of chronically painful conditions in obese individuals due to increased joint loading or upregulation of inflammatory pathways. However, conversely, another study showed decreased BMI among individuals struggling with opioid addiction. And also in individuals using opioids, there was a less prevalence of obesity in comparison to that expected for the general public. And this may be due to competing reward pathways, but both of these studies kind of underscore that interaction between obesity and opioid use. So my dissertation work focused primarily on noradrenergic signaling as a possible point of convergence between obesity and opioid abuse. So clinically, Lusamira was approved by the FDA to mitigate some of the symptoms of opioid withdrawal, and it works by acting on the alpha-2 adrenergic receptor. Norepinephrine is also involved in regulating food intake, and some of the FDA-approved weight loss medication, namely Contrive and Kisimia, suppress appetite by blocking norepinephrine reuptake. So one of the major producers of norepinephrine in the brain is in the locus coeruleus, which is a really small brainstem nuclei. The locus coeruleus expresses expression of dopamine beta hydroxylase, which converts dopamine to norepinephrine in the final step of norepinephrine synthesis. Although LC is a very small nuclei, it provides extensive projections throughout the neural axis. The LC has been shown since the late 1970s to be activated during morphine withdrawal. So in this graph, it shows naltrexone, injection of naltrexone resulted in hyperactivity of LC, and this hyperactivity of LC contributes to a lot of the withdrawal symptoms reported in individuals. The LC is also involved in feeding. It receives feeding-related projections from hypothalamic regions, and also LC is involved in maladaptive eating disorders, namely emotional eating, binge eating, as well as disinhibition, which is defined as a loss of control in feeding. Despite this evidence suggesting LC's role in maladaptive eating disorders, there hasn't really been any studies to date looking at the role of LC in obesity. So for my dissertation, we wanted to see some of the alterations in LC in individuals following obesity, and whether these obesity-related alterations affects LC responses to morphine. So the first part is to look at the role of LC in obesity. So we tried to mimic the genetics as well as the environmental exposure aspects of obesity. So we tried to mimic the genetic portion by using selectively bred obese-prone and obese-resistant rats. And then we fed them for 8 weeks on a 10% low-fat or 45% high-fat diet to mimic the more diet and environmental exposure aspect. We measured hemodynamic measurements, after which they were allowed 1 week of recovery, and we measured in vivo LC activity. We sacrificed the rats for histological confirmation of recording sites. So as expected, the obese-prone rats displayed an increase in body weight in comparison to the obese-resistant rats, and the obese prone that were fed high-fat diet showed greater body weight in comparison to all other groups, and this corresponds to the food intake data that we observed. With regards to hemodynamic measurements, we did not see any differences in blood pressure. With regards to heart rate, we saw a significant decrease in heart rate in the obese-prone rats. So next, we measured in vivo LC activity. So we anesthetized the rats using isoflurane anesthesia, and they were anesthetized through the duration of the recording period. We measured spontaneous activity for 3 minutes, followed by LC activity during sciatic nerve stimulation. So LC is characterized by its biphasic response to sciatic nerve stimulation. So right after the sciatic nerve stimulation, there is an increase in LC activity, followed by a period of inhibition, and then return to baseline. So we did the sciatic nerve stimulation every 5 seconds, and in these repeated 5-second intervals, we plotted the number of spikes in each 8-millisecond bins in these peristimulus histogram. So we define tonic activity as that 500 milliseconds prior to sciatic nerve stimulation, evoked activity as to the activity immediately following the sciatic nerve stimulation, and then for 104 seconds. So with our rats, we saw a significant increase in spontaneous activity in obese-prone rats, and this agrees with the increase in tonic activity we observe in the obese-prone rats. With regards to evoked activity, we saw a significant decrease in the obese-prone rats, and when we analyzed the signal-to-noise ratio, or evoked over tonic, we continually saw this decreased activity in the obese-prone rats, and we also saw a group times diet interaction effects, whereas the low-fat fed obese-resistant rats showed an increased activity in comparison to both of these obese-prone groups, and then an increase in the high-fat fed obese-resistant in comparison to the low-fat fed obese-prone rats. So given what we saw with LC activity and obesity, we next want to see whether these obesity-related alterations in LC activity can kind of mitigate LC activity following opioid withdrawal. So next we obtained Sprague Dawley rats at 4 weeks of age, and we fed them to the similar 10% low-fat or 45% high-fat diet. They were on the diet for an extended amount of time because this is when COVID happened. And then we subjected them to an escalating dose of morphine. So we give morphine twice daily using subcutaneous injections or saline. They underwent withdrawal for 2 days, followed by in vivo LC activity. Again, we histologically confirmed the recording site. As expected, the high-fat fed rats showed an increase in body weight and as well as food intake. During morphine dosing, those rats receiving morphine administration saw a significant decrease in body weight, and this was associated with a decrease in food intake in the morphine-treated rats. With regards to LC activity, we did not see any difference in spontaneous or tonic activity. And then with respect to evoked activity, we saw a significant increase with morphine withdrawal, but we only saw this effect in the low-fat fed rats, but this was absent in the high-fat fed rats, suggesting that there is some maybe protective effects of high-fat feeding in LC response to morphine. So during the dosing period, we saw a significant reduction in food intake and significant loss of body weights with morphine withdrawal. So we want to see whether this decrease in weight can kind of confounded some of the data that we saw. So next, we took Sprague Dawley rats and we age matched them to the previous group. And then one group of rats were ad libitum fed. The other group were food restricted to the amount that were eaten by the morphine treated rats. And then so we food restricted some of the rats, and as expected, as a result of food restriction, the rats showed a decrease in body weight. So for the LC activity, we saw that there is an increase in spontaneous activity following food restriction. There was a non-significant trend for increased tonic activity. There was no difference in evoked activity. And when we analyzed it to signal-to-noise ratio, there was a decrease in activity. So to summarize this up, so obese-resistant rats displayed increased sensory evoked LC activity, and this was exacerbated following low-fat feeding. Low-fat fed rats displayed increased sensory evoked LC activity following morphine withdrawal, but this effect was not observed in the high-fat fed rats. These changes in LC activity during morphine withdrawal was not attributable to decrease in food intake or body weight, but rather food restriction, increased spontaneous activity, trended for increased tonic activity, and decrease in signal-to-noise ratio. Some of the limitation of this study was that this experiment was only performed in females, it was only performed in males, so we don't know what the effects are in males. And also, we don't know whether differences in LC activity contributes to or results from differences in obesity susceptibility. So these are some of the data I generated during my dissertation. I'm really excited to work with Corinne and Jin Hao as part of my T32. So I'll continue to look at this intersection between food and addiction, but I'm making that transition from preclinical rodent work to more human side. So this is one of the projects that I'll be helping to work on, which is looking at the effects of BHB ketone esters on brain metabolism and alcohol consumption in alcohol use disorder. So alcohol use disorder, these showed low brain glucose, but increased brain acetate metabolism, and this shift in energy utilization during abstinence may contribute to withdrawal and craving. So previous work in their group have shown that ketogenic diets mitigate some of the withdrawal and cravings in alcohol use disorders. So next we want to see whether supplementation with exogenous ketones can improve brain energetics and reduce alcohol craving and intake. So we'll be doing MRIs and PET, which I'm really excited to get my hands dirty in. And then I'll also be working with Jin Hao on his grant looking at inter-individual differences in the prefrontal cortex and how that those differences can predict treatment outcomes in individuals with opioid use disorder undergoing treatment. So some of the risk factors that is proposed is gray matter volume, striatal connectivity, drug cue reactivity, inhibitory control, and self-evaluation. Well, some of that study is going to be going on. I'll also be analyzing data, previous data looking at whether BMI modulates naltrexone's effect on neuronal structure and functioning. Thank you so much for the opportunity to come to Penn. And I'm really excited to work with Corinne and Jin Hao. And I'll also like to acknowledge my previous lab for some of the data I generated. And I'll be happy to take any questions.
Cindy, thank you so much. And are you sure you've only been here since Friday? I'm teasing. So it was a wonderful tour through your thesis and a really exciting teaser for the work to come. I'll let other people ask questions. I know that other people have mentioned throughout the afternoon gender or sex effects. What are your thoughts about that, since you've been able to test one but not necessarily the other?
I think there are definitely going to be some sex effects. I think there are some studies showing that estrogen is involved in regulating the transcription of tyrosine hydroxylase, which is involved in dopamine and norepinephrine synthesis. So it'd be interesting to look at how obesity affect LC activity in females. I definitely think there are some effects.
All right. Any other folks have questions before we move to, I think, our final presentation of the afternoon, if you can believe it? Okay. I think Julie's going to do the honors of this special introduction for Julie,
Great,
aka
thank you.
Kate Brindleson. Go ahead.
Yeah, so thank you. And thank everyone for staying on the call. We realize without a wine break or a whining break or anything else in between, it's been a long afternoon, but we really appreciate everybody's input and the questions, and the presentations have been great. So our last speaker was a predoctoral T32 fellow, Kate Brindleson. She did her PhD work in my lab, and I'm gonna brag a little bit. She had 6 papers that she attributed to the T32 during her time in the lab for first author papers. So she was incredibly productive and started off with sort of a lot of preclinical work, but towards the end, her last project really involved looking at functional connectivity in mice over the course of opioid exposure, in a collaboration with Danny Bassett. And now in her postdoctoral fellowship, she's actually working in Danny's lab. And we thought that this work would be really relevant for the addiction training grant because she's now moved from opioids to alcohol. And she's going to tell us a little bit today about cognitive regulation of alcohol cue reactivity in a social context. So Kate, it is great to have you back for this talk. And you're there somewhere, I take it, because I see your slides.
Yes. Can everyone hear me and also see my slides?
Yes, looks good.
Yes.
Awesome. Okay. Thanks so much, Julie, for the kind introduction. I'm excited to tell you all today about some preliminary results on our study looking at cognitive regulation of alcohol cue reactivity. So prior work suggests that executive cognitive function plays a key role in modulating drinking behavior. And this is further evidenced by the observation that individuals with alcohol use disorder show deficits in cognitive regulation of cue-induced craving, suggesting that it's an important predictor of drinking behavior. And cognitive regulation strategies, such as focusing on the long-term consequences associated with substance use, may effectively attenuate cue reactivity by engaging prefrontal cortical downregulation of activity within craving-implicated subcortical regions like the ventral striatum. And so in this study, we wanted to address the question of how we might define brain states associated with cognitive regulation of alcohol cue reactivity and predict the input necessary to drive the brain to those states. So we set out to address this question by defining cognitive states in terms of an energy landscape, which represents all states that are accessible to the brain. And similar to the peaks in a mountain landscape, states that are further away are more difficult to access and require more control energy or input. So this schematic is a representation of the brain's energy landscape, where distinct brain states are defined according to the pattern of neuronal activity across the brain while engaging in a particular cognitive state or at rest. And the energy required to access a particular state depends on the brain's structural connectome or white matter connectivity. So one of the goals of network control theory applications to the brain is to be able to predict how we can reliably drive the brain to a particular state by providing some sort of control input, which could take various forms. So, for example, we could identify specific regions of the brain that strongly influence the control energy for state transitions and stimulate those regions using tools like transcranial magnetic stimulation to drive the brain to a particular state. Another approach could be to use a pharmaceutical intervention. And this is an example from a recent preprint showing that LSD can have a sort of flattening effect on the brain's energy landscape via 5-HT2A receptor agonism to allow the brain to more readily transition between different states. So in this case, rather than targeting specific brain regions to drive state transitions, we sort of lower the energetic barrier, making these states easier to reach. And finally, what I'm going to be focusing on today is how we might drive our brain to a particular state by enacting cognitive control. And recent work from the lab has shown that greater working memory load requires increased control input, suggesting that we could operationalize the control input necessary to drive state transitions in terms of cognitive effort. So in this study, we want to determine whether we can use network control theory to predict how regulating one's perspective while reacting to an alcohol cue will impact craving and ultimately how it might influence drinking behavior. So the data I'm going to tell you about today were collected from a subset of participants enrolled in a larger study called the Social and Health Impact of Network Effects Study, or SHINE, which is designed to provide insight into health behaviors and social interactions among young adults. So there are two major components to this part of the study. The first is a scan session in which resting state, task, and structural DWI imaging were acquired from the participants. And then immediately following the scan session, the participants engage in a 28-day period of ecological momentary assessment, or EMA, during which they report on their experiences of craving, drinking, mood, and other variables during their day-to-day lives. So during the task fMRI session, the participants react to images of alcoholic beverages. There are a total of 96 trials in which they are either instructed to react naturally, to take the perspective of a peer who drinks more than they do, or to take the perspective of a peer who drinks less than they do while reacting to the cue. So at the beginning of these perspective-taking trials, the participants are cued with the name of a person whose perspective they're supposed to take. After each natural react trial, the participants are asked to rate their level of craving on a scale from 1 to 5. And after each perspective-taking trial, they're asked to rate what they believe would be their peers' level of craving on a scale from 1 to 5. After the scan session, the participants are enrolled in a 28-day period of EMA. And during the EMA sampling period, They receive 2 text messages in the morning and evening each day, and are asked to report their level of alcohol craving on a scale from 1 to 100, as well as the number of alcoholic drinks they've consumed since the last survey. During 2 weeks of the 28-day period, the participants are asked to employ the same cognitive strategy that they used during the scan session, by imagining how their peer would react when they encounter alcohol cues, specifically a peer who drinks less than they do. And during the other 2 weeks, they're instructed to just react naturally to alcohol cues. So in the task, as I mentioned, participants are asked to rate their own craving or their peer's craving on a scale from 1 to 5. And so here, each of these points represents mean craving rating across trials for one participant. And we can see that most of the time when participants are asked to rate the perspective of someone who drinks more than they do, they rate that person's level of craving as being higher than their own, and vice versa, but not in every case. So the participants' perceptions of how much their peers drink and how much they crave alcohol are not perfectly correlated, suggesting that they may attribute their peers' drinking behavior to other factors as well. So next, in order to define the energetic cost associated with taking a peer's perspective while reacting to an alcohol cue, we use a linear model of neural dynamics over time. So in this model, XT gives the pattern of brain activity over time. So here I defined the initial and final states as the pattern of brain activity while a person is reacting naturally to a cue, and then while they're taking the perspective of a peer while reacting to a cue. A represents the structural connectome, which we derive from the diffusion weighted imaging. So it's represented by white matter streamline counts between every pair of brain regions in the network. And finally, BUT gives the control input. So the model that I'm using here assumes all brain regions are included in the control set, and in this case, we've parcellated the brain into a total of 400 cortical and 14 subcortical regions, using the Schaefer and Harvard-Oxford atlases. So in this figure, each point represents one person, and I've computed the log sum energy cost to transition from taking one's own perspective to that of a peer while reacting to a cue. And so what we see is that taking the perspective of a peer who drinks less is actually more energetically costly than taking the perspective of a peer who drinks more. So I next wanted to test the hypothesis that the difference in craving between oneself and a peer will predict the control energy required to take the perspective of that peer. And here again, each point represents data from each person, where the points are colored according to the trial type, whether they were asked to take the perspective of someone who drinks more or less. And this analysis suggests that taking the perspective of a peer who experiences less craving than you do is more energetically costly than taking the perspective of someone who drinks more than you do, irrespective of condition, because as I mentioned before, participants don't always rate the craving level of their peer as being in the same direction as that peer's level of drinking. Okay, so to briefly summarize, we see that participants in this study rate the alcohol craving of their peers who drink more than they do or less than they do as different from their own, and that perceived peer craving and drinking are positively correlated. Taking the perspective of a peer who drinks less is more energetically costly than taking the perspective of a peer who drinks more while responding to an alcohol cue. And the difference in alcohol craving between oneself and a peer is predictive of the amount of control energy required to take that peer's perspective. Okay, so now I want to talk about what I want to do next. So moving forward, I'm interested in determining which regions and which connections drive the difference that we see in total control energy for driving transitions to a higher or lower craving state. So my hypothesis is that craving implicated subcortical regions are going to be more influential in driving the brain to a higher craving state than a lower craving state, and that engaging this circuitry is less energetically costly than engaging regions that are involved in inhibitory cortical control. So we can assess the role of specific regions or cognitive systems by suppressing activity within individual regions and then observing the impact on the total control energy for state transitions. We can also assess the role of specific connections by varying the strength of individual edges in a person's structural connectome. So by computationally increasing or decreasing the strength of a connection and then asking what the impact is on total control energy. Another means of addressing this potentially would be to derive effective connectivity networks from these data. These would give us a measure of causal interactions between regions. So the edges in this case would be weighted according to their utilization rather than the density of white matter connecting them. And this approach could also potentially tell us something about the direction of activity flow through the network. Right now, I'm also working on constructing multi-level models that we could use to assess whether we can use control energy to predict craving and drinking behavior in the real world, accounting for variables like exposure to alcohol cues, which participants also report on in the EMA surveys. Finally, I think it would be really interesting to be able to relate a person's ability to engage in cognitive control to their social network. So one of the really unique features of this study is that we have EMA data both from individuals who were scanned, which is data I showed you today, as well as from peers within their social network. So this figure sort of illustrates how we might embed a person's brain network within their social network and ask how the behavior of their peers and relationships with those peers impact their energy landscape and their drinking behavior. Okay, with that, this project represents a collaborative effort between 5 different labs, 3 of which are at Penn, 1 at Columbia and 1 at UNC Chapel Hill. I'd like to especially thank my postdoc mentor, Danny, the other PIs involved in the project, and members of each of their labs that I've primarily worked with, as well as our funding from the Army Research Office. And thank you so much to Julie and Anna Rose for inviting me back to talk today.
Great. That was
fun.
I was going to say, I think at the end of this, seeing all of those beer mugs, I think you could have found a lot of craving in our attendees today. But I just had a real quick question. So this idea that it's higher energy to predict or to have the perspective of those that drink less. These subjects did not have alcohol use disorder diagnoses, correct? These are just normal controls?
That's right. Yeah, they're healthy controls. Yeah.
Interesting. Is there a plan to look at folks with alcohol use disorder to see if that's any different or no?
Potentially, yes. So I think like I mentioned, the really unique feature of this data set is that we have the possibility to bring in social network variables as well, which we could only really do by including college student participants in the study. But then I think this provides a really nice opportunity to develop tools that we could then potentially apply to data from other populations. So I think that would be a long-term goal.
I'm just going to say, Kate, it's really fun to see these elegant network tools applied to phenomena that are sort of near and dear to our hearts. It's very fun for me to see that, and it would be really fun. I mean, you could even with your college students, you'll have a continuum of people who are going to have at this point binge drinking that will progress to something that they don't manage. It's very tricky in college because there's a lot of heavy drinking that doesn't progress to alcohol use disorder. So one of the things that you could turn that into, though, as a strength, is that you could see in a cohort whether or not these network tools are strong predictors of people who are going to get into trouble. Is there a vulnerability signature, which is always where my head is, I guess, even when the behavior looks kind of similar. Like there's a lot of heavy drinking, but not everyone is the same. Not everyone has the same genetics. Not everyone has the same other vulnerabilities, the epigenetic vulnerabilities. And some people are going to come out of college and do just fine, even though they had a lot of heavy drinking. And other people are going to progress to a significant problem. So, I mean, there's a million things you can do with this, but since you have your foothold in the college population, I don't even know the SHINE cohorts. Are they all college students, basically?
Yeah, they are from Penn and Columbia.
Gotcha. And of course, no better guide than Kevin Oxner for reappraisal. But I think, at least with our just folks who are really struggling with the disorder and extremists, it is difficult. It is, even without an elegant graph, it's energy requiring for them and very difficult to maintain an inhibitory or top-down kind of control relationship between the cortical regions and, for example, the amygdala. And so we've been able to show that people who were able to do that for several minutes in a connectivity kind of study, but not as elegant as this, just regular old, one region to the next kind of connectivity, driven by the a priori hypothesis, that the people who were able to do that, to establish that inverse relationship between the dorsal cingulate and the amygdala, went on to, quote unquote, likely better outcomes. But the people who were unable to establish that even, right, those regions weren't even talking to each other, they weren't connected well, had the phenotype that is known to go on to poor outcomes. So that sort of thing, including things that we do with Q activity to try to predict outcome just straight up, regular old activation and differential activation and activation under conditions of attempted inhibition, to see what's the real best predictor, because we sort of have our sort of head to the ground trying to get better predictors so that